Well, I don't need my slides to start and say thank you to Julieta for inviting me to speak here today and for putting me first so I can relax and enjoy the rest of the day. I'm going to talk to you about the Pentatomoidea of Florida. This is something I've been sort of working on for a long time. It started when I came to Florida in 1981 and soon after met Dick Baranowski down at Homestead. Dick, who's now deceased, how long it's been, asked me to help him with a project on the Pentatomoids of Florida, much like the Coryidea of Florida that he and Jim Slater put out. At the time, the Arthropods of Florida series required you to list all the label data from every specimen you looked at. And that got to be an overwhelming task. And I just finally said, nah, can't do it. And Dick didn't pick it up either. So the project kind of floundered for a while. And then I started teaching. Lyle Buss asked me to teach Pentatomid identification in his class. USDA asked me to do a training session. So started developing keys for the Florida fauna. And Lyle over the years has helped me with it. Susan Halbert has been very good about running the keys and telling me where I've screwed up and making suggestions for improvements. And then in the, I think it was 2011 or 2011 or 12, Julio Medal and Andy Santa Cruz started working for the Florida Department of Agriculture, looking at a host range study for Chusocus japonicus, which is a samurai wasp, which is parasitoid of brown marmorated stink bug. And so in order to do this, they had to rear a lot of bugs to get the eggs they needed to test. And so, they came up with a lot of information from the rearing and a lot of pictures of immature stages that have not been published previously. So there's a lot of good information there, and Julio put together a manuscript, but because this wasn't a rearing study, this was just kind of a sideline. The data wasn't very consistent, and there was it was hard to put into publication. It just didn't flow. And nothing happened. It didn't get published. And then in 2020, I think Kate Fairbanks picked up responsibility for getting this information out, and again asked me to join her, and it has floundered ever since until last year. Paul Skelly asked me that we publish a Pentatomoidea of Florida, and then we could incorporate the rearing information where it was appropriate, and the pictures were appropriate. It didn't have to be consistent across species, and it'd be a good way to get this information out. Paul promised me some technical assistance in terms of somebody taking pictures for me, and that's Ashley Sturgill now, who works for Susan. And so the project is going forward. We're about halfway done to putting this together. It'll be published as occasional paper of the Florida State Collection of Arthropods. Is that right? So appreciate Paul's help and everybody else's help in getting this thing going. Just a little history. There have been lots of papers, as you might guess, on different Pentatomoid groups within Florida. But overall publications, Barber was the first in 1914 with Hemiptera, Florida. I'm going to talk a little bit about each one of these on the next slide. So Banduzzi in 1917 published a catalog. Its first catalog devoted to American North America, American North of Mexico. Blatchley came up with a guide to the Heteroptera of Eastern North America. He had keys and descriptions and drawings of a lot of the species. This was really a good work. Torrey Bueno put forth a synopsis of the Heteroptera of North America, and this was the first time that we had keys for all the fauna north of Mexico. And as such, he was not really a catalog, it was more of a key. And he didn't always include Florida in the distribution of some of these things. So he kind of missed a few things that were in Florida. Then Froschner, the latest, 1988, he and Tom Henry published a catalog of the Heteroptera, and Froschner did all of the Pentatomoid carton. So if we look at sort of the number of species level taxa, I say that because there are a couple of subspecies that occur in Florida. both subspecies. So Barber came up in 1914 with a little under 80 species. Now Van Duzie's catalog was really well thought of, but in reality, he missed some things that Barber had, and he actually added a few things that I don't think belong here. So his catalog had about a little over 60 species. Blatchley actually described a number of things from Florida and got the species up to a little over 80, 85 or so. Torre Bueno, as I mentioned, didn't always include Florida in the distribution, so the numbers dropped off a little. And then Froschner's work added about, about 20, 25 species to our fauna. Currently, we're at 124, and that's with taking 12 away from Froschner because I don't think they occur here. So, so we've really had quite a few things since 1988. Come on. 124 taxa, 24 of these are immigrants, and I won't, the few are invasive, but I'm using the term immigrant for just anything that's foreign. And then since Froschner, we've got 7 that I call hidden taxa. They were either here and we didn't identify them as such, or they've been collected. They were in the southeastern US, Georgia, Alabama. We just didn't pick them up here. So added 7 there. I think there's 12 that should be thrown out. And these are, to me, as important as immigrants, because once they appear in the literature, they're persistent in the literature. Somebody's got to throw these things out. And it's frustrating to have a bunch of things that you don't occur here. So the affinities, we have 4 endemic taxa. 2 of these are subspecies. We have probably the most in common with the southeastern United States. Most of our species, a lot of them have relationships there, followed by the southwestern US. And you might expect that because southeast is close to it. And nothing separating us, no barrier. Southwest, a lot of things have in the southwest we share with the southeast and as well. And then neotropics and West Indies. And there are a number of things that are found in all of these geographies. You look at genera, you got about 51 genera, and we share about 4 with Europe or the old world. Everything else is New World as well, with about the same range. I'm going to talk about immigrants, and I'm going to talk about things that need to be removed if I have enough time. Looking at immigration here, prior to 1925, so whole history before that, we've identified 3 species that probably were immigrants at that time. So not very many, really. 25 to 50, 1925, 1950, only 1. Starts to pick up 51 to 75, we had 3 immigrants. 1975 to 2000, we had 8. And after 2000, we had 9. So you can see the trajectory of immigration is going up. And I don't see any reason to suspect that it won't continue. So I expect more immigrant species, which makes it important to know what we have here now. So where did these immigrants come from? Well, the lion's share came from the Caribbean, which again would be expected. We probably already shared everything with the Southeast for the most part. So new things coming in through the Caribbean, a pretty high percentage. Western US, kind of surprisingly, but I think maybe with global warming, some of the Southwest things have gone a little further north and come over. We'll talk about that. A few things from Central and South America that have really come either through the Caribbean or Southwestern US. And then we've got 4 Asian things, 3 of which have come in the last 15 years, which is probably indicative of the fact that we're getting more trade with Asia and expect to see more and more things coming from Asia. All right, where did they land? Where'd they show up? Where were they detected? Talk about Asia first. Most of you are probably familiar with this critter down here. On the lower right, Hemiptera, a brown marmorated stink bug. It's a pest arrived in the mid-Atlantic states, and I think we picked it up many times before it became established here. So we think it's coming down. It's a home invader in the fall, and when the snowbirds pack up their mobile homes and come down here, we figure it's coming down on 75. Pardon me?
No.
It's coming down on 75 and 95 with people coming down here. It didn't establish at first, we don't think. But eventually it slowly progressed further south, and now we have established populations. Megacopter carberia came in, this is a kudzu bug, came into Athens, Georgia area, and it feeds on kudzu. And it's it also came through corridors of travel, but really kind of spread throughout the kudzu growing area and just gradually crept down. And then about 2012 came into Florida. Our most recent one is this guy here, Brachyplatus subaeneus. It was found through an iNaturalist posting by a student here at UF. And it came into Miami Beach. Somebody took a picture of it there and posted it on iNaturalist, and we sent inspectors out, and they confirmed the presence of this bug, sent a specimen. It actually showed up in Panama first, and then moved to the Caribbean, and I came here through the Caribbean. So its occurrence in Miami kind of fits with the other Caribbean thing coming in. All of these are pests and have potential to be pests, so pretty bad pests. Hemiptera is one of the worst. Here are the Caribbeans, and I don't have all of them, pictures of all of them there, but it's pretty interesting. Things from the Caribbean all show up in Miami and the Keys and in southeastern Florida. And you have a lot of you have a lot of ports down there, and a lot of boats come from the Caribbean and dock there at these ports. And I think that's how a lot of these things are coming in. We know, like, for example, with termites, we get a lot of termites that way from the Caribbean and even South and Central America. The Western immigrants, they're kind of a mixed bag. We had a couple here. This is Euschistus quadrator and Homamus proteus, which I think came across, just moved across. Quadrator was picked up first in Tallahassee, and proteus in Gainesville. So these are things that kind of just came across. But then the others, we've got Rhytidophorus compactus, which showed up in my swimming pool, first one collected. Frank Mead caught this critter, Basidia, on the hood of his car at a meeting in St. Pete back in the 90s, and we never found it again. We looked and looked for it, never found it again. It's showing up on iNaturalist, and we've got specimens from St. Pete. Contacted the young lady who posted some of that information. So, and then we've got Hylospilus, which is showed up over here, and it's a Western species, but it's been spreading somewhat in the Caribbean, and I suspect it came through the Caribbean rather than across from the western part of the U.S. I'll talk briefly about some species removed that I think should be removed from our fauna. This is Neophornis fimbriatus. Banduzzi described this thing from a specimen sent to him by Torre Bueno, and it was from Florida, but without a definite location. It's not here. We've never seen another one. There's been no reports of this thing. This is a Cuban bug, and it has affinities with several other genera in Cuba and Hispaniola. This thing doesn't belong here. It needs to go, even though Florida is a tight locality for it. There are a number of things that are found in the north that have been reported from Florida, much of this in old literature, some of it in fairly recent literature. Just as variolarius and cervus are very similar in appearance. Variolarius is found in the north, but I don't think it's found in Florida. It's just the cervus is common in Florida. You can see they look very much alike. The males are easy to tell apart because the Variolarius has a dark spot on the genital cup, and there's no dark spot with cervus. But the females are hard to tell apart, and we rely on these little dark spots at the end of on the edge of the abdomen that are variable. So as a result, females of cervus a lot of times get called Variolarius Florida. 34 seconds. Okay. So anyway, these are misidentifications, and I think they need to go. There's one other that I won't have time to talk about. So basically, fauna is changing. I think the immigrants are going to continue to come in and impact us, so we need to know what's here now. I think some of the uncommon southeastern species will eventually show up in Florida, and we need to pay more attention, or I do, to citizen science, because using that we can detect things a lot quicker, I think, than just relying on ourselves to collect in this small network of people. So with that, I think I'm done. Thank you.
Thank you, Joe. Thank you, Joe, for this incredible talk. I'm waiting for the out.
Working on
it. Does anybody has one question for Joe?
I'm also opening up the chat, see if there are questions in the chat.
Joe,
Yes.
immigrant equals established or no?
Yeah. And I would call the Bennett something that just comes in.
You have another question?
I'm removing, you meant removing from the list, not removing from the state.
No, we're moving from our fauna, from what we call the fauna of Florida.
Yeah.
You have another question?
With clarification. Were those 50 grams new or totally sliding over from previous?
Oh, no, it's new.
New.
Yeah.
Each histogram.
Yeah, you talk about the immigrants. Yeah. So, yeah. It's increasing pretty dramatically.
Thank you, Joe, again. We now have Matthew Moore that is presenting collaborative efforts. And the title is Cosmopolitan Parasitoids, Mining Biodiversity Databases Reveals Unexpected Abundance of Intercontinental Distribution in Platygastroids Wasps. So you got the speaker here, and the stage is yours.
Fantastic, thank you. First of all, I've got to thank the co-authors on this paper. Without their contributions, none of these data would make any sense. Okay, I'm unable to provide any context to these DNA sequences without their input. This is very relevant, and I'm actually glad I followed Joe here because the movement of Hemiptera and specifically Pentatomoids around the world is very relevant to this talk. So a little bit of background, as Joe brought up, invasive Pentatomoids are becoming a big issue in the United States. And we have some case studies here from really the last decade where we had the kudzu bug move in, brown marmorated stink bug, and then Bagrata bug. There was a classical biological control agent identified for each. Those were brought into quarantine, but before they were approved for release, the parasitoids showed up in the wild anyway. I was personally involved with this last case here, Gryon ethereum, aptly named the Gryon from the ether. This species was not on the radar at all, and then all of a sudden it was all over the world. So this is kind of alarming, completely circumventing our regulatory processes. And it's not just classic biological control agents. At the same time, we learned about Gryon ethereum, we learned about 6 Sciatoceps. So 6 is an old world genus. There was one species that was known to be inventive already in the United States, and we found a second, Sciatoceps, reared from these pest stink bugs in Florida. Here's its distribution in the state. It's already widespread. We found out by going back and looking at old specimens, it had been here at least since 2018 and completely unrecognized. CO1 sequences from our Florida specimens, 100% matches to South Africa, whatever that means. Here's the wasp, very beautiful. And it's not just our lab group that's noticed this pattern. So Telenomus remus is a biological control agent that's been used for fall armyworm. It was recently being proposed that it would be released in Africa to control this species here. It was already there. Okay, you're getting the theme here. And it's not just platygastroids that are showing this pattern. Oencertus nizari was a second parasitoid of kudzu bug that showed up in Alabama in 2019. 2018, this was published. It's subsequently been found in Florida. We were involved with that. Importantly, though, it was not the same CO1 variant. So there's 2 different populations. Whether this is indicative of a multiple introduction event or just a single event that was diverse, we don't know. But the parasitoids are clearly following their hosts, their invasive hosts around the world. And it gets crazier. There's trophic interactions here. Okay, so this was another project our group was involved in. Acroclossoides sinicus, this is a pteromalid. You can see some variable egg damage here in the middle. Eggs that have the hyperparasitoid have a different chewing exit hole. This is this green guy is the wasp here. Acroclossoides sinicus was an old world species that we now understand to have whole Arctic distribution. There was a synonym that was described from the United States, but by looking at specimens that emerged from Trissolcus parasitoides Hemiptera eggs, and sequences from around the world, we realized, oh, this is a single organism. So we synonymized the North American one. Now we understand hyperparasitoid is the same thing that occurs all around the world, coming out of our biocontrol agents. All right, so it's really wild. It's getting wild out there. You can ask yourself, well, how many cases are there of this? Very difficult question to answer. The US Geological Service, the Simpson et al. 2021 study, they made a compilation of what they verified as adventive species in the lower 48, Alaska and Hawaii. And they say 29 platygastroids adventive in the US. Based on our current research, we think this has to be an underestimate. In Europe, there's only 8 species that are listed. Now, some of these are also intentional releases, but then some aren't, but only 8 in Europe. No way, that's a huge underestimate. Other people have looked at this with morphological data. So this is a good species. They call them tramps, global tramps, identified morphologically 3 genera. This graph here is from that paper, this map, excuse me. These yellow points in Australia and New Zealand were the original distribution. Specimens were later found across Australia, and then Hawaii and California. So that's not a natural distribution. Humans are mediating that. It has to be the case, right? So I had a bright idea. We need to mine biodiversity databases to find hits of this pattern and then work backwards to try to identify the organisms, because it's clear that we're like on a treadmill and we're falling behind. Like we're not up on what we should be able, the descriptions of these things moving around. We're just not finding them quickly enough. Barcode of Life database, through 2 national initiatives in Germany and Canada, there are now millions and millions of sequences for animals, and a couple million for insects, and a lot of them are not identified. They're only identified to maybe family. So there's tons of data and not a lot of resolution. So in our case group, platygastroids, this is very apparent. There's 140,000 available sequences. Only 128,000 of them are not identified at the species level. So almost no species IDs, and even 108,000 are not identified to genus. Okay, so there's lots of data, not a lot of identifications. So our approach was to identify bins, which are sequence clusters or centroids in the database that have been calculated for very similar sequences. They don't necessarily equal species, okay, but they might be parts of species lineages, or if there's other evolutionary processes, 2 species might be represented in a bin, but they're close, they're approximations, okay? We found all of those bins displaying intercontinental distributions, because we thought those are likely inventive. We identified as many of them to genus as possible using the infrastructure from the database. We compared hits in GBIF for the same pattern as sort of a morphological, like, baseline comparison set to the molecular data. And then we added sequences from Florida to improve resolution in the database, but also fish for intercontinental hits. I would mention we recently sent to the BOLD administrators 2,500 genus level identifications from our Hymenoptera group, with many fascinating taxa in there. Very, very good progress. So here's what it looks like in the database. This is an intercontinental bin. It was unidentified at the family level, Cosmopolitan. This Brachypterus female here, he ID'd it as genus Baetis. It's got occurrence points in Norway, Germany, Sweden, and Canada. We know that this genus are parasitoids of spider eggs. So on the database, they give you a lot of calculations about what's in the bin, how many members, how the diversity of the distances of the sequences. And then you can pull all of that data off yourself, which is what I did. And then I searched for exact matches across large geography distance. Here's another great example, unidentified coelionid, Xenomeris from the images. This would be a continental record if this holds up. This is an old world genus that's never been recorded before. The Canadian record here is from British Columbia. It should be followed up on, but if that's true, yeah, it's a new record for the whole genus in the hemisphere. Again, Norway, Germany, and Canada. Genus is a parasitoid of crab eggs. We're very excited about this one, or at least I am. Unidentified platygastroid, we ID'd it as Eustenotis. Germany, Belarus, and Canada are the occurrence points. Now, there is a species of Eustenotis that is used as augmentative biocontrol for a wheat midge, the history of which is kind of convoluted. The identity of the organism is kind of, but it's been released in Canada in the late 80s and early 90s. It was in their subsequent surveys, they didn't really find it, but then the species was reported in Montana in 2015. I suspect that this is the parasitoid. It's just not easy to ID, but we're going to try to follow that up as well. So overall, we found 153 bins with intercontinental distributions in the database. We had to provide genus level IDs for 100 of them. Otherwise, they were just a family. The breakdown of the genera, 10 in platygastroid, 15 in Coelionidea. Most of the bins were relatively small. So that's what this chart here shows. A huge proportion of the bins had under 82 members, and then the larger bins that had like many, many hundreds of sequences, those were rare. We found 50 bins with exact CO1 matches across continents, highly indicative of an actual transplantation there, not a natural distribution. There are many inconclusive matches, some where there's missing data at either ends of the sequences where I said, it looks like an exact match, but there's some variation that I can't account for. So we'll call it inconclusive for now, but maybe it really is another exact match. Canada and Germany are the top localities for this pattern. So here's, these next few charts show a neighbor joining tree of all the 8,500 intercontinental bin sequences. This is great story. I mean, Grynotherium, okay, was collected in Pakistan, taken to California in quarantine. We found sequences that matched it in Mexico, then Egypt and South Africa. While we are writing the paper describing the species, people sent us specimens reared from BMSP in Chile. They said, hey, what's this wasp? And we're like, oh, isn't that amazing? We know exactly what it is, but we're the only ones in the world who know what it is. You're very lucky you sent it to us. Here's another example. Unidentified platygaster species. These are parasitoids on gall midges. The colored lines here indicate exact matches. So we've got matches in Central Europe, all the way in the Russian Far East, then also in Canada. We have no idea what this species is. This is a fascinating one. So some of our traps in Jacksonville have turned up really interesting hits. This one, okay, Port of Jacksonville, really good match to Southern Australia. So no way this is a natural distribution. Telenomus has many hundreds of species in it that haven't been revised. We don't know what this wasp is. We probably won't for a very long time, but we know that it's moved around the world. Kind of a cool pattern. Our morphological data set was based on those same genera we found in the Barcode of Life database. We queried on GBIF. This is an example of the Trisulcus data and where the distribution points are. The GBIF data are pulled from many different sources. We then got all of the data off there and then sorted through the occurrence points, just like we did with bullet data, to look for species that have been reported on multiple continents. We verified those hits in the literature as much as we could. We flagged dubious records, potential mis-IDs, things of that nature. So the GBIF and the literature data set is similar in some ways, but quite a bit different than the DNA data set and others. So for example, The GBIF data set has 24 genera and 118 species, so a little bit less diversity than the molecular data set, but they don't quite overlap at the generic level. So we have 4 genera that were only detected via DNA, 6 genera that are only known as morphology literature records. So there's some gap there that maybe can be targeted by researchers to fill, so we can get this whole picture. And there were some interesting species levels discrepancies as well that kind of surprised me. Telenomus, which is hyper diverse but has relatively fewer revisionary works. We had plus 23 DNA hits compared to the morphology. So Telenomus is moving all over the world, but we really don't know what those are. We had 17 fewer hits for Trissolcus, which is a very closely related genus to Telenomus, but has many regional revisions. So that's more documented in the literature. Just a phone call. No, that's not phone. For the future, what we plan on doing is trying to follow up with these identifications. We have specimens from the German barcode of life that have matched the Canadian specimens. We have someone in Germany looking at those now. We're going to try to put names on as many of those as we can. We think that this severely complicates regional revisions. That's right. If you have this fauna that's moving all over the world, you might describe something as a new species in one place, but it's just going to clearly be a synonym in the future, right? If you're not comparing globally, it's going to be really hard. We think that for classical biological control problems using egg parasitoids like these, you have to have max force species identification upfront, or greatly risk missing the fact that your parasitoid is already in the environment that you want to put it in. That seems to be the big takeaway, is that if you have an invasive pest, if your parasitoid agent is an egg parasitoid, there's a good chance it's just going to follow right behind under your nose. We think in total, if we rectify the data sets right now, we have maybe 30 genera and 200 species with this pattern. Determining whether they're naturally whole Arctic or transplants that are human mediated is a difficult question. There may be some approaches that can elucidate this in the future, and that would be very fruitful future research. This one we're excited about. We want to scale up the approach. So everything we did with this group was basically manually done and verified, but we want to mine the whole database for Insecta. So we have a script that we are working on. It's almost ready to try, where we're going to blast the whole barcode of life database and pull up every intercontinental bin from Insecta, and then provide the bins to the community and say, okay, Hemipterists, you should look at these bins, see if this makes any sense or not. And we're excited for that. And then hopefully by February or early March, we will submit a manuscript on this, and you'll be able to read about it in the future. But we're excited about the results. We think it really informs at least a DPI's mission of trying to identify these faunas and things as new records as they move in. We really lack a global baseline for most groups. This might be a great way to try to push that forward, to establish that baseline. Is to look through these biodiversity databases for hyperdiverse groups and start pulling out relevant information. And with that, my talk's over. Thank you very much.
Thank you, Matt. This is an amazing talk. Fantastic.
It was great to follow Joe. It's super relevant. I mean, it's just flows right into it.
Yes, it is.
We have time for a few questions. Yeah.
I wonder why Germany and Canada came up so high on your, is it just a matter of more people sampling there?
Yeah, that's where the most dense sampling is. It's the result of national initiatives, plus also really they're the most globally connected, right? So a lot of those trade routes are going to be same. The organisms that are moving, we have a history of colonization between Europe and Canada and United States. It's all, yeah, it's just probably the best two ways that they're connected.
It seems that makes all these biological control programs questionable. If the parasitoid is there already, and still they don't have control unless you can exert control by augmenting the population,
Sure.
you know? It's a problem.
Yeah.
What's the script that you mentioned at the end? What is it based on? Is the
Like the programming environment. One of our pathologists is at DPI is doing it. It's something he's running through R. I can't tell you exactly.
But so you downloaded old,
Yeah, we downloaded the data set for Insecta. And we identified all the occurrence points, so that was important, and we had to assign them to a continent. Which is complicated for Russia and then some Papuan areas. And then, yeah, we're almost ready to pull the trigger, see how many there are in Insecta.
thank you again, Matt.
Yes.
Great, thank you. And now we have Andrew Johnson, who's gonna talk about the new records and taxonomic changes of Scolytinae in Florida. Johnson, you got the speaker there, and the stage is yours. Perfect. You've got 15 minutes.
All right, just give me a second to start the screen share.
Yeah.
Okay, so hello everyone, I'm Andrew Johnson. I'm going to be talking about the bark and ambrosia beetles in Florida. I'm actually going to kind of include Platygastroid in the numbers, which are in addition to Scolytinae. So the bark and ambrosia beetles of Florida, there are a few species that are very well known and have either a recent history of causing problems or a long history of being a known pest. But there's actually a lot of other species. And this talk will kind of go over my efforts to kind of make the fauna of Florida and kind of keep up to date with what's known there. And there are a lot of species. This is from a poster, which has taken me over 8 years to compile all the known species. It's very close to being there. There's a few tweaks needed, but as you can see, there's a lot of species there. And before I get into too much detail, the fauna of Florida can change in a few different ways. The kind of easiest ones are the new species and new records, whether they're described from here. But then there's kind of more complicated things where taxonomic changes cause them to be added or lost. And that's kind of a bit more messy. And it really needs like the concept of that record, which is often not recorded. There's also unpublished and published, which is debatable if that's a record at all, if it's unpublished. And this talk includes some of the unpublished ones. So just as a nice visual representation, this is over the last 23 years. And you can see it's increased dramatically. At the turn of the millennium, it was just over 140 species were known from Florida. And now it's over 170 species. So there's some new species, and these are kind of the easiest ones to imagine. This is how I started working on this group, was my PhD, and I was working on the former Griffalini. And there are a couple of species which are probably native, very kind of rarely collected, overlooked, which are described. Then additionally, there's a couple of species which were described more recently, and these are species which are in Asia, but also here. Actually, the one at the end, I collected the holotype of it. But it was described from Florida, but it has just last year been recorded from Asia now. So we can kind of plot this on the accumulation. There have been really big trapping schemes in Florida for wood borers, particularly EDR and CAPS, which some people here might be involved with. And this is a really big scheme where they're trapping, often targeting particular pests, but it also records novel records for Florida. And these records are a mixture of species that are pretty common in the eastern North America. And we've just got a few records finally, whether they're ephemeral populations or not is unclear. But then there's a few other introduced species which have kind of expanded their range into Florida, which have been detected. As well as a few new continental records as well. So you can plot those on the chart as well. Some of them are really difficult to identify. These are 2 records that were kind of in the collection, which were kind of unnoticed, which are just undetermined. And luckily, Don Bright described them just as I got to look at them and managed to identify them as two additional new continental records. And actually, this small one, Big Mare Boris, that one's actually been found really widely, like all the way to southern Brazil and everywhere in between. So it's actually a really common species. It was only described 3 years ago, really broadly distributed. Yeah, we can plot these in this chart. It's not all from these trapping schemes. This was in Fort DeSoto near St. Pete, and I was just on vacation, and this is a sea hibiscus, a non-native shrub. And looking at the twigs, I kind of recognized this little beetle, and I knew exactly the genus, and it's not known in North America. It's a genus in South Africa and Asia only. And yeah, this turns out to be Rhinoceros parvulus, which was the first record in continental North America. There was another species described, which is actually synonymized, which is in the Caribbean as well, which is recently found in Puerto Rico. It's only known from sea hibiscus. Then going on to Cyclopidion again, I mentioned one was described. This one here was described. Just a few years ago, there's only one recorded from Florida, but since several have been found, and this is only published last year, and these are all really, really similar. There's very subtle differences. They're kind of pseudocryptic species, so if you look really carefully, you can find differences, but you really have to look. But then, just last year, I got another one just light trapping again. I saw the first live specimen and recognized it while it was alive. It's pretty remarkable. And this has got a beautiful sulcate declivity. It's actually a really distinctive Cyclopidion. And this one's going to be published soon. It's in press. And this one is a really broad distribution in Asia, all the way from Thailand all the way up to Russia. So it's probably going to do pretty well here. It's probably not going to be a pest. We can add these as more recent additions. So these are just what's been collected and actually published, but actually they're collected before they're published, obviously, and there's going to be a small lag. And actually, we're just keeping up. We catch up, but there's more and more collected all the time. And actually, the number of species isn't quite there yet. And there's some which are kind of unpublished. This one was from 28 years ago, where there's a single specimen collected in a nursery. It wasn't imported material, it was like grown there. And it's an unusual species that feeds on the flowering stalks of bromeliads. I wouldn't be surprised if it's established. It's not where people actually look, and they probably don't go to any traps. There's another Hypothalamus. It's kind of another one that was recently described. The kind of Hypothalamus are really tricky, so this one's just been overlooked and was misidentified for decades. So then, just want to mention, I mentioned before how taxonomic changes the more complicated one. And these kind of are much more difficult to track, and it's kind of difficult to release records for these. For example, there is another Monathrum species, which are kind of a interesting, smooth-looking cortholine. And Monathrum mali is really common here. And there's another species in the Caribbean that's currently a synonym. I'm convinced it's a different species. It's got a slightly different declivity, much smaller, and it's very consistent change. And we have a single specimen collected in the Keys a couple of years ago. The specimen on the right is from the Bahamas, a female. Hypothalamus is a notorious genus that's really difficult, and there has been some taxonomic changes which I disagree with, such as Hypothalamus javanus and brunaeus. They're recognized as synonyms, but they're kind of clearly different if you can see the top of the fronts. This is often obscured in pin specimens, but it's I've also got some genetic evidence to corroborate this. Another species was Hypothalamus peramanus, which was reported as not being in Florida recently. I disagree with that because they kind of, to me, they look really different to the species they're identified, which is Hypothalamus erectus on the right there. And again, this one's actually most easily diagnosed by the cuticle on the top of the head. There's a few other small differences and genetic data as well. There's also some which are kind of other complicated cases, like this one. Celebrinus attenuatus was recently recorded in Florida as part of the EDRR trapping scheme. But actually, this is not correct. This is a misidentification, so we need to take that off the record. But then there is this other one, which is this smaller one. This is Celebrinus taxonomic. And then there's a smaller one, which this one is identical to specimens of Celebrinus subgranulatus in Asia. So it should be recorded here. The problem is these are pretty extreme examples, but they're very difficult to distinguish. They're good genetically. But this one's actually probably the most common emergent beetle, which is kind of funny. But to accept this, if you look at at least CO1 sequences, to accept this would render Saxocynae paraphyletic. So the most common one, we have both clades in Florida, so that would be kind of a messy thing. So I'm kind of being cautious with this record. Xyleborus species, these are notoriously difficult as well. There's one which looks like perfrans, which isn't, and kind of working on what it is. It's probably a native species. It's rarely collected, but widely distributed in the southeast. And then Xyleborus ferruginous and Xyleborus bispinatus, they were notoriously difficult, and we were wondering if they were synonyms. And then a student, Gabe LeMay, has done a good piece of work to show that they're actually distinct, both morphologically and genetically, but there is some overlap in the morphology, so it's not always diagnosable. Coccotriepes, there's at least one new species. I really don't want to describe it, but I'm running out of museums to check. Then there's another Coccotriepes, a global species, Coccotriepes cyperi. Here's one boring into a mango. They're not often recorded as pests, but they're really common on fallen fruit. I think they wait till it's like a little bit rotten, like beyond marketable, so they're not really an economic pest. But this one, again, Coccotriepes cyperi, the one that is most common is not Coccotriepes cyperi. And Coccotriepes cyperi is actually pretty rare. I've seen only one specimen. And when I went to look for it, I found another one, which is also kind of different and like a little bit different. And just with CO1 phylogeny, which I know isn't perfect, especially for Scolytinae, but they do come out in very distinct clusters that are already known from elsewhere around the world. So this is kind of an annoying species complex, which I know there's 3 species, but we can't really deal with it. So that's how I've contributed to that's the contribution so far. So really we have these like collected and published, and there's a slight lag, but really there's another line somewhere which we don't really know what it is, but it's how many there actually is. And that's what we really need to get to. And it's probably increasing. There's probably new records or new species arriving. And that's what we need to really work on. I mean, this lag isn't good. It is just taking too long. We shouldn't really wait 28 years to publish a record. And that's what I'll be working on over the next few years in a few different projects that all address this issue. Just completing the taxonomic work. I'm going to be the EDRR sorter for Florida, which is going to really speed up the detection and everything. I'm always working to digitize and working with databases. I really like database structures and stuff. So if anyone's talking about that, I'll be happy to. And also we're kind of developing methods for faster processing using AI or image processing techniques. So hopefully we can really close this gap. So with that, I've got lots of acknowledgements. Lots of people already helped out with these taxonomic changes and identifications. And I'll take any questions.
Do we have any question for Andrew?
So what determines the host specificity for those species?
It varies, it depends. Some are really generalist and some are not. But actually recording host is difficult because people often record a host as they found it on a plant. But sometimes they're kind of messy records. Sometimes it's like they were beating a plant and it fell out, which is not really a host record, or they hung a trap on that tree. There's lots of messy records.
But for example, if you have avocado tree that was hit by frost and then you have
absolutely,
is it always the same beetle? Or
The avocado is almost entirely generalist species. There's no specialist avocado species there. Most of the ambrosia beetles that attack like is garden and stuff are like the really generalist ones that feed on every plant.
David Serrano would like to know if that poster will be available.
I really hope so. It's actually, I need to reformat it. I had the text too small, and to make it legible, it needs to be like 6 meters wide. So I need to remake it. If anyone's got a good way to print it nicely, like a really good quality printing source, I'd be interested to know that too.
Any other question? All right.
If
Thank
I
you.
may, I would love to ask folks to, I don't know who is here involved in the PR or perhaps programs. But how do we, how much of a issue is it the fact that we have this lag in systematics of lights, no problem at all to it's a dramatic issue for the program. Does anybody know? Anybody come up with that? That's a good question, actually. What's the
It's the early detection, rapid response. So it's supposed to be quick, but it's ideally to spot things that are known threats. So they can actually do eradication schemes, which only really work if you get there really quickly.
Thank you.
Thank you. Now I invite to the stage Andrei Sourakov, probing Lepidoptera diversity with wing pattern manipulation experiments. It's going to have 30 minutes, and you got the speaker, you got the thing, and good luck to you.
Thank you. Yeah, so to lighten things up a little bit, I'll talk about something very different, show you a lot of pictures of colorful butterfly wings, but I'll try to make it relevant to taxonomy somewhat. The talk reflects my latest obsession with phenotypic plasticity of Lepidoptera wing patterns, and I've been interested in Lepidoptera all my life, so understandably was attracted to it, like many people, through the diversity and beauty of their wing color patterns, which are used quite extensively in taxonomy, of course, because there's certain thing that jumps to it, and frequently they're good characters in Lepidoptera wings. But for the purpose of this talk, what I want to attract your attention to is that these 4 butterflies on the left all represent the same species, actually a bordered patch butterfly from the same population. So there's tremendous variability. All these butterflies in this drawer, African swallowtails, also represent the same species. Right? Males can be, or they all look like this, and males in some parts of Africa also just look, females in parts of Africa look like males. In other parts of Africa, these butterflies mimic different milkweed butterflies. So tremendous ability to adapt. These butterflies actually can represent identical genotypes, not just phenotypes, right? So different phenotypes, but they are determined by the conditions in which caterpillars are developing. So this is polyphenism, example of polyphenism in European map butterfly. And this, for example, butterflies represent different species, not necessarily closely related, so they quickly converged on similar color pattern in the effort to mimic each other. So butterflies can change their appearance relatively quickly in the course of evolution. Comes, for example, a model toxic species in the area where black and white butterflies occur, and all of a sudden, a butterfly that looks like this begins to look like this or this, right? So these changes can happen very quickly, and there's not very much genetic divergence necessary. In fact, these butterflies can form hybrids among themselves. So how do butterflies do that? So clearly, wing phenotypes, they're very useful in taxonomy, but they do not represent different species, even though they may represent genetic diversity sometimes within a species. Frequently, however, unlike most groups that you study, we have people coming from all walks of life to Lepidoptera taxonomy, because many people collect butterflies and moths, they're members of amateur and professional Lepidoptera societies, and they tend to publish frequently taxonomic papers, and frequently using wing patterns as characters. So if you come across, for example, a butterfly that is quite common normally, has very familiar to all stable phenotype like Gulf fritillary, but you find something like this, you may choose not to name it because it's clearly a case of aberration, especially if they're just one or two specimens like this lying among a large population. But if, say, you are collecting in the tropics, right, where butterflies and moths are known much less, and Altomeris, different species of Altomeris come to light occasionally, and one moth like this comes, well, there's a question, right? What do you do with it? Is it a different form? Is it an aberration? Or is it a different species? And frequently, these things get named as new taxa. So there's a lot of work being published currently on genetics of butterfly wing patterns, and I'm not going to go in this talk into this very dense woods, but most of this work, I have to say, is done on model species that are easy to maintain in the lab, such as painted lady butterfly, and people study in depth how different genes are responsible for wing pattern formation. There's another genus of butterflies, Heliconias, long wings. They form this amazing mimicry rings, extremely plastic phenotypes. So all these are the same species, and you can see how they follow each other in mimicry rings. So this is another model that people have learned a lot about. In fact, I was inspired to do the work presented today by the talk that was given here by Arnold Martin, now a professor at George Washington University, then postdoc in Patel's lab in Berkeley, who was talking about his work on vintage gene ligands in Heliconias. And during this talk, something struck me is that you can actually manipulate wings of butterflies and moths, and don't have to use fancy, difficult techniques like CRISPR. You can use pharmaceuticals to inject butterflies or moths during the formation of wing pattern and change the wing pattern, specifically the vintage positive wing pattern elements that tend to expand or contract in response to this or that chemical. So two questions immediately when I heard this talk popped into my mind. Can we understand a little more about homology of individual wing pattern elements? And also, more relevant to this meeting, can we understand a little more about diversity of phenotypes by applying these treatments to a wider variety of species? So by homology, we all understand what it means, right? So you see this eye spots in different groups of insects and different Lepidoptera, but intuitively we do understand that these are not homologous organs, right? That they're homoplasies, they are formed by different developmental mechanisms, and but they serve similar function of repelling predators. And by the way, there has been long-standing efforts to understand homologous organs in butterfly wing patterns. They were based on nymphelid butterflies, and in fact, the term nymphelid ground plan was coined as long ago as 100 years by Eger, 1924. It has been developing ever since. So under this nymphelid ground plan, all butterfly wing elements belong to one or another symmetry system. In nymphelids, exemplified here by a satyrine, they are represented by the series of bands that concentrically, imagine this as being a center, concentrically are distributed through the butterfly wings. But of course, it doesn't describe the tremendous diversity of butterfly and moth wing patterns out there beyond nymphalids. And for example, if you take these two relatively common moth species that we have here in Gainesville, the iron moth and the polyphemus moth, they have both these eye spots located on hind wing in the same place, but are these organs homologous or not? They actually use them in a slightly different way. Iron moth covers them like this when they at rest, and then when they disturb, they bring their forewings up, and the polyphemus moth actually rests like butterfly with wings closed, and they flick their wings open. They belong to different subfamilies separated by tens of millions of years of evolution. So you can see here the eye spot in Saturnus usually is located on this M2, M3 vein as a center. And actually, if you look closer at these elements, you will see that the eye spots also formed by a series of scales that different elements within eye spot actually are formed by scales of different shapes. So if you apply, again, you can see closer these eye spots here, you can see that their similarities are somewhat superficial. What I've decided to do is apply some heparin treatment, which is sulfated polysaccharide. Many people are familiar with it as it's used as a drug for treating thrombosis, coincidentally. You have to inject a forming pupa in first probably 10 to 20 hours after pupation. And so I looked at the way the eye spots have changed on this moth. And you can see that actually different partner elements within eye spots have changed in different ways. So some of the elements probably are homologous and some are not. Closer look at this treatment. So this answers the question, by the way, about this Colombian specimen published in Lemaire's book, collected in 1967, that is clearly very similar aberration to the one that was obtained in this study. So this also helps to explain some of the variations that you see naturally throughout North America in the polyphemus moth, quite variable species. In fact, some of the populations are described as different species. So pandemics come, and this seems to be like an ideal set of projects. You can rear lots of caterpillars at home, and so I decided to kind of diversify this technique into a bunch of different species locally occurring here in Florida. And it involves rearing hundreds of caterpillars, monitoring them, etc. So again, probably without pandemics here, I probably wouldn't have done that. But start with the Bella moth, one of my favorites actually. I've been working with it for the last 10 years on different aspects of its chemical ecology. But what I was noticing is that frequently during the colder times, you see that moth are kind of lighter. Again, these were anecdotal observations. And in fact, 7 years ago, I decided to try out the cold as a phenotype-inducing characteristic or parameter, and in fact found that they indeed become lighter and darker, both lighter forewings and darker hindwings under influence changes in temperature just 7 degrees. So here's a good example of phenotypic plasticity, temperature dependent. Heparin actually replicated to some extent this treatment, showing that probably Wnt genes are involved in control of these characters. In monarchs, the changes were very different from normal nymphalid ground plant, but monarch certainly is a very strange, unique species, and it belongs to a very basal lineage of nymphalids that has been split from nymphalids about 60 million years ago. So, and again, they are migratory, they are mimetic. So there are many reasons why their wing pattern may respond very differently to heparin treatment. But in certain populations, the residence populations throughout the Caribbean, you can see similar darkening of the wing margins in monarchs. And on the underside, this character naturally is rarely found in Danaus flexicus, but may be found in some of its relatives in the genus Danaus. Here's the common buckeye. On the left, you see the natural population here, and on the right is a heparin-induced phenotypes. You can see the deletion of orange bar and loss of definition along the margins, and also reduction in the size of eye spots as heparin concentrations increase. The changes that can be obtained, and here's the underside. And why it's of interest for taxonomy, there are a number of taxa out there of Junonia that share characteristics with the phenotypes that were obtained using heparin. Here's the Asteracampa clyton, the Tony emperor, and these are heparin-induced phenotypes that I have not encountered in the collections. This is the underside. Yeah, this heparin-induced. This reminds me a little bit of the changes that occurred due to seasonal plasticity in some saccharines, like Melanitis leather here. This is the wet season form, and this is a dry season form. You can see a similar reduction in eye spots and loss of definition in the wing pattern. This is the Vanessa Atalanta, the admiral, and this is the heparin-induced phenotype. You can see that the expansion of the wingtail positive wing pattern elements actually does not depend on the color of the elements. In this case, the orange and blue and white areas are expanding. And I found this specimen in the collection from 1957. Clearly, somebody experimented with cold effects on European admirals, but produced very similar phenotypes to the ones produced using heparin. Here's well familiar to Gulf fritillary. This is a gradient of heparin-induced phenotypes that was obtained, and this is the underside. What's interesting here is that the silver spots that intuitively one would put as homologous organs, right? If you think of each spot as an organ. But actually, it turns out there are convergences. There are wingtail positive elements here along the margins and wingtail negative ones closer to the base. So color, shape, position on the wing, actually all of those characteristics can be quite misleading. But this helps to explain those rare aberrations that we see in the collection. In fact, out of 1,000 specimens that we have in the collection, we have only one aberration like this and one like this. So this is not a common occurrence, but maybe some cold-induced phenotypes can be obtained like that. But you can see expansion and contraction of those organs. And again, I'm referring to wing pattern elements here as organs, but that's what they are. I mean, butterflies use them for different functions. And you can see how variable they are in close relatives of Gulf fritillary. Here's one moth who yet gave me some caterpillars, which I think here. This is mulberry leaf tear, and I took advantage of those caterpillars. And obtain some transformations here, which are kind of interesting because you can see some variation in these characters in related species, the same genus. Here's well familiar to you state butterfly, the zebra longwing, and the gradient of phenotypes obtained using heparin. In fact, I came across photographs of similar phenotypes. They're very rare, but they do occur. So here's some swallowtails, spicebush giant, and Patus polydamus. Again, not going too much into the swallowtails. Here's the changes. They are very reminiscent, actually, the heparin phenotypes of the phenotype of Heraclitus taurus in Costa Rica. And the Patus polydamus changes are quite convincingly resembled by populations of subspecies Christopheranus on St. Kitts Islands. So finally, the last species I worked with, and it's the first lycinid species on which any wing pattern manipulation work has been ever done, is the Atala butterfly. So lycinids have their wing patterns arranged in concentrical circles like this, but they are very different from nymphalids. They split from nymphalids with nymphalids in the course of evolution, probably about 50 million years ago. So people have tried to quantify their wing patterns to come up with terminology, but they resisted overall trying to draw any parallels with nymphaliid ground plant, knowing how the far divergence of these two families goes. So here the heparin obtained phenotypes here, and they can actually link you a little bit to the relatives of Atala butterflies that we have in Mexico and the Caribbean, and also can help explain a very rare, in fact, I only seen photograph of on iNaturalist of a single individual like this from 4 years ago on Miami. We don't have anything like that in the collection, as far as I know. So, but from the point of view of interest, kind of more general interest, using this technique, I was able to make some analogies between the nymphaliid ground plan here, the bands, and the bands of lycinids by looking at how individual bands expanded or contracted. So hopefully I stayed on time.
Absolutely. Thank you, Andrei. We got a lot of time for questions. I got one question or an observation. So all these changes in the phenotype questions a little bit the functionality of or the adaptive value of these characters. Do you know any data why this is changing so much? Because let's assume it's avoiding predators, right? If you're reducing the spots, you're not avoiding predators. So it's dependent maybe on the cold, but you're not avoiding predators.
Right. So you're referring, for example, to seasonal, right? Polyphenism in saturnines. So in these butterflies, there's an interesting correlation between behavior and the patterns, and also hormones and reproductive activities. So, for example, during wet season, the saturnines that exhibit a kind of very bright, attractive pattern, they're very active, they mate, and females immediately lay eggs. And so they need these defenses. During the dry season, when females emerge, they're ready to mate, but they don't produce any eggs. So they go into hiding and they stay very still. There's very little access to nectar. And so their behavior actually correlates very well with their phenotype.
Lovely. Fantastic. Interesting. Thank you. Any other question?
Are the difference in color pattern related in addition to temperature or season? The host?
No,
No.
I have not. Anecdotally, people say that, but I think it's because they use different hosts during different seasons. And they have observed it. In all my experience with Lepidoptera rearing for some over 40 years, I have not encountered any correlation between hosts and genotypes.
I asked because in springtails that is the case. They feed on something, they're one color, they feed on something else.
No, I have not encountered it, and I don't think any records of that are accurate as far as I know. However, that said, caterpillars of geometric moth can actually reflect the substrate on which they occur by the color of their skin. So they have cells that are sensitive to light and they can change the color. But that's the only example that I know. And the same is true for pupa as well of, say, swallowtails.
Any other questions? All right, thank you, Andrei. Now we have Aswaj Punnath.
Yeah, you can call me Ash.
Aswaj
Yeah.
Punnath. Fantastic. It's a collaborative effort, but he's going to present morphological convergence in repeatedly evolving ecomorphs of avian feather lice revealed by nano-computed tomography. Good luck for you. Got the speaker, you got everything.
Yep.
Fantastic.
Just give me a second to set the screen,
Yes.
please.
You got about 15 minutes. Whew.
Hi everyone, I'm Aswaj Punnath, PhD student in the entomology department of University of Florida. I'm actually a myrmecologist and ant person, but this is another project I've been working on since the first 2 semesters of my graduate program. Morphological convergence in repeatedly evolving ecomorphs of avian feather lice, and we are using the novel imaging technique, computer tomography, for this one. We all are familiar with lice, right? At least the human head louse, Pediculus humanus. I'm not going to ask how many of you have still have the irritation with the lice. But today I'm going to talk about a different lineage, the bird lice that you can see here. This is one of the recent paper published in Nature Ecology and Evolution. One of my co-authors published this one. So you can see here the bird lice come here, like different species are there. So Superfamily Ichneumonoidea. So I'm talking about they belong to the order Phthiraptera. So they are permanent ectoparasites. They spend their entire life cycle on a host, on a bird host. And we can call them obligate ectoparasites because they need the host for their survival. So we all know how much irritation or difficulties these lice will cause when they have, right? When they have high infestation. So it will lead to significant feather damage, which eventually goes to lower ability to thermoregulate in the host organisms and secondary infections that will eventually lead to death of the host. So if lice is there or parasite is there, then the host will develop some kind of adaptation to overcome this difficulty, right? So in birds, there are 2 main defenses: preening with the bill and scratching with the feet. Just like we scratch our head to remove the lice, birds have these amazing stretchers. Some birds have, you can see this, comb-like stretchers on their feet that facilitate removal of the parasite from their head. So if the host has this kind of adaptation, I'm pretty sure like the lice will have some kind of counter adaptations to overcome this. Yes, lice have similar kind of counter adaptations, and they evolved into different ecomorphs. So what are ecomorphs? They are local variety of a species whose appearance is determined by its ecological environment. One familiar example is Darwin's finches. We all know about it, right? Based on their microhabitat specializations, some organisms will develop different morphological adaptations. So another example is ecomorphs of anomalous lizards. So one good example for this one is islands, where the area is very limited. So organisms will have some kind of adaptations to overcome the competition between species. Similar thing happens here when it comes to birds. You can see different ecomorphs of feather lice. Based on the place where they reside, you can see head, those in the head lice, those in the body lice, those in the wings, wing lice, and there are some generalists. You can see some kind of morphological differences between these organisms, these different ecomorphs, right? Hair lice will be referred to hair lice. You can see their head is somewhat triangular shape, and their temporal region is somewhat broad, right? But when it comes to wing lice, their body is long and slender, and they have long limbs. Body lice have a similar structure, but they are kindly smaller, but their head is not that big as head lice. And we have some generalists with different variations in their shape and structure. So there are many studies on avian feather lice, mostly by Dr. Kevin P. Johnson. So previous studies shows a repeater morphological divergence within host groups and convergent evolution across feather louse lineages. The importance of the present study, this is the first detailed study characterizing and quantifying the morphological features. And as I said before, we are using the nano-computed tomography for this one because we will get the 3D aspects of shape. So we will generating more volumetric data and visualizing morphological features in a 3D framework. Coming to the hypothesis. So based on the morphological characters that I mentioned before, like the head lice have the bigger temple region and other kind of stuff. So we are assuming two main features that are believed to be beneficial for parasites, the lice, to counter the adaptations of, counter the defense of host organisms, the bird. So one is that head lice will have the largest mandibular muscularization. I told you before, like they have the temporal region, long temporal region. So that shows that they might have a largest proportional volume of muscles. So we are quantifying this proportional volume of chewing muscles to compare across different ecomorphs. And I told you about the wing lice, which has a longer body and having kind of like longer limbs. So we are also measuring the proportional length of leg parts and between more mobile and sedentary ecomorphs and quantify differences in tibia and femur length. So we collected 89 specimens from 62 species of lice, and most of the collections are postmortem either state fumication, and if it's live birds, we are testing with pyrethrum powder. So the collected specimens are like preserved in alcohol. So we are CT scanning them in Phoenix machine and X-ray machine. We have this facility in the nanoscale process facility in the campus, and we are with the CT scan images. We are doing the body segmentation in VG Studio Max software. This is how we are performing this kind of CT scanning. So you can see here, I mounted different lices here, like because I'm using a tape method for that, so that will have a low density material. So I will keep the stage here. I mean the sample here, and the source will produce X-ray and be edited to capture the images. So the volumetric and measurement data, we are measuring the proportional volume of chewing muscles, and also the length of femur and tibia individually and together, and also the proportional length values, that is the femur plus tibia length divided by the width of metanotum. And we are testing statistically to see the significance of the data. We are also doing the geometric morphometric analysis of the head shape. You can see different landmarks here, and using the geomorph package and doing the generalized probabilistic analysis using geomorph package and the principal common analysis to look at the differences in the head shape between different ecomorphs. And we are also considering genomic, the phylogeny using genomic sequences of lice based on target set of 2,395 single copy nuclear genes. Coming to the results and discussion. Yes, as I mentioned before, there is a significant difference in the proportional volume of chewing muscles across different feather lice ecomorphs. So the head lice have the largest and wing lice the smallest. You can see here, wing, generalized body and head. Head lice have the largest proportional volume of chewing muscle compared to other things. So you might ask me why head lice having this larger proportional volume of chewing muscle, right? So this is an adaptation. This is called an adaptation to escape from the host defense. I told you before, like birds, they will always try to scratch their head and remove the head lice, right? So the head lice wants to bite down and grip onto the feather barbs to avoid being removed by scratching. So that's why they have to facilitate the mandibles. They should have a stronger mandibles. So that could be the reason for having a larger proportional volume of chewing muscles. Well, we couldn't find any differences across different sex of the specimen did not have a significant impact that you can see here when we're comparing across different ecomorphs. Coming to the proportional length of the legs between ecomorph groups, we found a significant difference, as I mentioned before. So the wing and generalist have, as well as the wing and body, wing and head lice have significant differences in their length of their limbs. So again, we will ask the same question: why wing lice having a longer limbs, right? There is an interesting behavior here. So most of the wing lice, they are involved in forces. They move from one host to another host with the help of hippoposid flies. See, hippoposid flies. So when hippoposid flies are coming, this wing lice will clasp their legs and hold onto their body. So that will facilitate forwarding movement, the movement from one host to another host. So that's why one reason long limbs in wing lice. So long limbs help to grab the legs of hippoposid flies and facilitate forces. So you will have a question like, what about other ecomorphs? So we found like head lice are also involved in forwarding movement with the help of hippoposid flies, but they're using their mandibles for grasping to the body of hippoposid, not the legs. So we found significant differences in the head shape also. You can see like it's distinctly separated in the morphospace. But the generalist comes in between, it's in the intermediate region. So generalists also overlapping with each of the other three groups. This is the phylogenetic relationship between different ecomorphs. And apart from this, we are doing some kind of analysis to see whether what is the effect when species, different ecomorphs are sharing with another. I mean, like different species are co-inhabiting a same microhabitat, what will happen? So we are also performing those analyses now. Coming to the conclusion, the nano CT scan data are useful tool for assessing unique morphological differences in very small inset. You can see here, we are getting the 3D aspects of its morphological characters, right? Compared to the traditional method. So we are getting 3D aspects of the shape, including dorsal and ventral points. Coming to the bird lice, there is a repeated evolution of morphological characters based on the microhabitat specialization. They have similar morphological characters. Each ecomorphs have similar morphological characters. So there's a convergence in overall morphology, and it's a result of the same specific morphological transitions for different lineages of the same ecomorph. And I would like to thank National Science Foundation for supporting this project, Nanoscale Research Facility, Florida Museum of Natural History, AFS Entomology Department, and my Lucky Lab.
Thank you, Ash, for the amazing images and presentation. Has anyone has some questions for Ash?
How does a louse know that it's on the head and not?
Yeah, that's an interesting question. I don't have a perfect answer for that one, but yeah, we'll see that one.
There's a question. Oh, right. Do head and body lice also use hypoglossids for raising?
Yes, they do.
Fantastic.
But the way they attach the body is different. head lice uses their mandibles, while the wing lice they use their legs for attachment.
Pelicans have lice that live in the gullet. What, you happen to know what guild they fit into or ecotype? Are they considered head ones, but it's like a different type of microhabitat?
Yeah, so this is you mean like the different ecomorphs differentiation, right?
Yeah, exactly.
So this is purely based on the morphological observations. So I'm pretty not sure like how much this data will be useful for differentiating other similar kind of like bird lices. But we found like there are exceptional cases. Some of the ecomorphs are not falling into any of this category. Some of them are showing like intermediate regions.
Any other questions?
All right, thank you.
Thank you, Ash. So it's me, the next one. So I'm going to talk about the male genitalia in Ichneumonoidea, I hope.
Yeah, I'm still doing the switch the screens.
So it's going to be about 3 hours. And I'm going to provide some of what we are finishing right now. So it's just me presenting, but it's sort of collaborative efforts, and I will present the my collaborators by the end. And I'm going to show you some images. I'm going to talk about why genitalia usually are useful. I'm going to move on what we know in Hymenoptera, what we know in Ichneumonoidea, and what we have done so far to try to create a unified terminology. So we all know the fascination that entomologists have for male genitalia in insects. This is the word by Nano. It's dissecting out the genitalia of previously unstudied specimen is like unwrapping Christmas presents. And it is still true for taxonomists. In fact, genitalia and taxonomy are used in Lepidoptera. They're used widely in Dipterans, and they're used, of course, also in Coleoptera, in which in some groups have been pushed towards the exploration of the endophallus, so the not the sclerotized part, but the more conjunctiva part of the genitalia. But also we know that, let's say in phylogeny or in evolution, they're extremely important because we know that genitalic features are usually have a strongly phylogenetic signal than non-genitalic features, at least in some groups. We know that they are important to study sexual evolution, the shapes of it, how does it work? And then, of course, for comparative anatomies or simple study of the musculature, like in this case, what's happening with the semitid, why do they, how they come to evolve this type of features, morphs of the traumatic insemination. But what about genitalia in Hymenoptera? What do we know about it? We know a little. There has been study done. This is one of the first modern study almost 20 years ago in the Hymenoptera. Thanks to Schulmeister, we were able to study the basal across Hymenoptera later, but at the beginning, toward the basal part, the symphyta. They are still used right now in some work. But what about other groups? We know, working with Phthiraptera, they are widely used. This is a comparative anatomy, but they're still used in also species description. And we have some data for ants, recent work by Brandon Bodinu. Okay, so it seems to be pretty good. But what about Ichneumonoidea? What about this huge group with 2 families, 80 more subfamilies, 40,000 and more species worldwide, distribution worldwide, parasitoids, so extremely important also for ecology. And also, as you see, they're extremely fascinating from the perspective of external morphology. So what do we know about those, the genitalia of these guys? Well, we know very little. In fact, these are the work done so far on the Ichneumonoidea. Peck, 1937, with 2 works. Then those are for Ichneumonoidea. Then we got Pratt, 1939. And that's it for the Ichneumonoidea. And the last one for Ichneumonoidea, in this case, Braconidae, in 1952. And that's about it. We have the Schulmeister that I saw, but it's very general, only 2 specimens. Nothing else has been done. Why so? And this was the big $1 million questions. Well, we have just 4 elements in the male genitalia of Ichneumoneids. And right now I'm gonna refer just to the Ichneumoneids because that's what I'm working mostly, but this is of course useful also for Braconids. There are just 4 elements. We got a cupula, which is that highlighted parts in the genitalia, which on the side views you can see can be also expanded laterally. Then we have a gonostyle, which is the area external to the genitalia, which in some group within the Hymenoptera can be divided in a basal and an apical part, sclerite. So this can be considered a cluster of sclerite that in Ichneumonid becomes only one. We don't see any separation in that area. Then we have a middle part, which seems to be unique in Hymenoptera, which is called the Volsella. It's not present in beetle. And if we do a section of it and we look inside, so internally this way, we can open it up and we'll see 2 sclerites, possibly. This one here, which is the gonosiculus, and the basal one, which is the parasiculus. This one also will be divided into different areas. I'm not going to talk about that. And then we got the penis valva, sometimes called Edeagus, but the Edeagus also include the endophallus. Right now, we're just talking about sclerites. And so the penis valva also can be divided into two different areas, sort of distal one and a proximal one. And proximal is in, I think, about closer to the body, all right? So it seems to be pretty easy to define those 4 elements. However, despite having just 4 elements, we have multiple names. And I think this one is one of the reasons why we have such troubles working with this genitalia. So for instance, I give you some example. So this is the cupula, and those are all the names that has been used in cupula since, well, I think 1800, all right? And 21 names, and you can see this fantastic name, horny ring, for this part. And this is just one sclerite, pretty easy. But let's take the gonostyle that I told you before in Ichneumoneids is usually one single sclerite, but in some groups can be divided. So the single sclerites, or the area, let's put it this way, can be called in this way depending on the author. If we have 2 different sclerites, the basal part has all that names throughout history, and the apical one has all these. So you don't have just 21 names for the area, you have 21 names plus 16 plus 11, I think, or 12, that was before. And this has created what I call, and not just synonyms, but we have also homonyms. So this is the case of paramer. This is what paramer for some authors is. This is paramer for what for some authors. So only the apical part of the gonostyle. For some others, the entire part of the gonostyle. For some others, the gonostyle plus the volsella. For some others, the entire stuff without the cupula, which is this little part here. So everything except the cupula. What we're going to do about it? All this synonyms and homonyms are created a terminological nightmare. And this is me trying to figure out this morphology and the male genitalia behind me, all right? So this has started just with a simple exploration and took me almost 2 years to figure it out, and with the help of many others. And that's why I think that a unified terminology is essential in Ichneumonoidea, and I would say across Hymenoptera. For what? Communication, first of all, so we know what we are talking about. To assess homologies in the future, for comparative and phylogenetic studies, we want to explore this stuff and see how they fit into a broader perspective. And of course, to assess the phenotypic variability across taxa, as well as pushing the morphology into what is called the phenomic era. Otherwise, we're gonna always lag behind phylogenomic, and we're not gonna get there, and morphology will become possibly irrelevant if we continue like this, right? This has been done in at least one group in the Drosophila melanogaster. They did that, so they create a standardized nomenclature. But also this is slowly being doing also for other parts within Hymenoptera, in this case, the mesopleuron structures, all right? So what we did is, I take all the studies, I compiled a synonymic list, give you the concepts, the definition with the HAO, a sort of discussion of why we're eliminating some of these terms and what should be the preferred term, the terminology, since we're talking about Ichneumonoidea, specifically in Ichneumonoidea, and eventually, if there are some parts of morphological variation, with the hope that this one could be used for future studies in comparative anatomies. But also, since we're talking about a very complex structures, there are also muscle within, so I'm giving you an alignment of all the muscles from back in the days. I'm mostly using the most common work, the biggest, let's put it this way, with what should be the preferred terminology under label and what single authors use as symbols or name for that single sclera. Muscle. And then, of course, I'm also, since we're talking about Ichneumonoidea, I'm taking the work with the Ichneumonoidea and giving you where different authors have found present absence of those muscles. There are some inconsistencies. Look at this one. We don't know why. Possibly we need to better assess it with the comparative anatomy. I'm not giving you any description for the moment because I think it's too much. It's already 40 pages, and I lost almost half my life doing this. So what we have found, though, and why it should be interesting, well, first of all, because they're extremely beautiful, and you can find a lot of characters that nobody has ever assessed. This is, I'm going across subfamilies here. So this is the Cremastine. You can see it's pretty much a normal genitalia for Hymenoptera. We found this muscle, which is the interpenis valva muscle that helps opening and closing the two penis valva, possibly to help into the reproductive part. We don't know exactly what they're happening, but they are associated with a midline separation of the two penis valvae. And this one has been reported in the basal Hymenoptera and by Schulmeister in Ichneumonoidea, never been imaged. We found it, it's present, and it's across Ichneumonoidea. And this is the last part here, letter C. You can see a section of the genitalia. You can see all the muscles inside. Those are the abbreviation for the muscle. If you need, we can talk about that later. But let's go on. The mesochorine, the gonostyle, is extremely reduced. You can see a sort of, the muscle are extremely compact. They are present no matter the dimension of the gonostyle. And in Pomenine, I think it's fascinating, just recently imaged, we found this structure here. So I told you before that in Ichneumonoids and Braconids, we have a single sclerite, not divided by two sclerites. So you don't see an apical portion, apical sclerite. But you see this one in some groups have never been reported in any work that I've seen so far. And this is a conjunctiva-like structure. So it's less sclerotized. What I'm thinking is, because you got muscle here that you can see that goes from the basal part to the apical part, then I think that possibly this gives more flexibility to the apical part of the gonoforceps and the gonostyle. So at this point, possibly they invented another way to move the apical portion of this structure. And also we were able to re-identify some of the features that were present in one group. I sort of lied to you. There is one genus in which genitalia are sort of consistently used, but they're not very easy to work with. And this is the genitalia of this big specimen. And they were talking about, this is our other images that we are focusing on the apical part. They're talking about a pad and a brace. These are nothing, these names are not used anywhere else than this genus. What we're thinking is that this structure here, it's present also in other Hymenoptera, and it's possibly used to transfer sperms into the female, because you see the small penis here, it's very short. So possibly when they insert the penis, the pad helps in transferring the sperm towards the female, since the penis is very small. All right, we're not sure, it's just an hypothesis for the moment. We still are not sure what this is. We're hoping to see it's a gonosquama, something like that. We're not sure what the brace is, if it is a sclerotization. But this will be put out with the paper and maybe inform future researcher and see what happened. And with this one, I would like to thank my collaborators, my supervisor, and the people at the UCF that helped me using their confocal laser scanning. And yeah, that's it. I hope you enjoyed. And if you have any questions, I'm here. Any question for Davide? Yes.
So the pad and the brace,
Yeah.
I was trying to make out what was exactly happening. Is the pad coming off of the gonostyle? It's part of the gonostyle?
Yeah, I think so. It seems to be continuous with the gonostyle, and it's continuous with the brace. Okay, so the brace goes into this sort of conjunctiva structure, all right? But yes.
And it just like kind of lines up with the penis?
Yeah, it seems so. But I haven't gone across Triphonine, which this genus belongs to, nor across the entire Natalia group. So it's possible that somewhere it's detached at some point. We don't know, but it's fascinating. There are some structures similar at the apical part of the gonostyle in Hymenoptera, but they're not the same thing. They're completely different. They're used for suction cup. In this case, it's not a suction cup. It's a completely different thing. Okay.
I have a
Yeah.
question. Do you think it's possible to use genitalia to get to species level, or they are just distinct for subfamilies?
I don't know. It's according to Pack and Pratt, possibly yes to species, at least to some groups, but it's never been tested. I think that possibly to at least the families and possibly to genus. We need to test it and go across groups. Could be. People say no, but I don't trust CSE data. Yes, sir.
I find your images quite amazing.
Thanks, sir.
So you said it's laser confocal?
It's the confocal laser scanning. It's been used quite a while in insects, never been widely applied though. It's what they use in biomed science. You don't need to stain anything, you just bleach it, and then you pump up the laser 97%, and they're not happy, but I'm gonna do it. And you can see the contrast. So these are not colored stuff. They are the way that they come. And you have different density, muscle and sclerites, and then there are some parts in conjunctiva that you can stain in blue, but it takes more time. So since I have an old confocal, a single image took me almost half an hour to have a very high resolution. But with the new one, you can also 15 minutes, 10 minutes, depending. Possibly in the future will become easier. And they are storing glycerol, so that's pretty good. You just put in there and then.
So the colors that we're seeing are fluorescent?
Yes,
Autofluorescent?
autofluorescence, yes. All autofluorescent. I can go back. Can I go back?
Yes.
There you go. Yes, those are all autofluorescence. You can see the blue here, the dappy. There you go. Yeah, it's a contrast. Any other questions for me? All right, I think we are getting to the break, the lunch break. I invite everyone to stay here. I think that Julieta already prepared everything, almost. And so I invite you to take a break, and then we're gonna reconvene here at 12:30. Also, you guys in the Zoom, I'll see you.
And welcome back. I hope you enjoyed your lunch. So we will have a few talk in the afternoon, and we are having a coffee break of 15 minutes around 2. As a reminder, we are always recording the talks. So if you don't want your talk to be recorded, just let us know. And for the presenters, I will give an heads up when we have 3 minutes left and 1 minute left. Okay, for those who want to participate for the dinner tonight, please give your name to Rieta so she can reserve a table for us. And now I'm gonna introduce the first speaker, Erick Rodriguez. And his talk is fieldwork of Anastrepa Lepidoptera, mainly in the Western Amazon. The stage is yours.
Thank you. Good afternoon, everybody. Thank you for coming here. Listen to this. My main focus will be about what I have been doing for the last 10 years, at least, in Central and South America. So first, I would like to introduce my colleagues that are working with me right now, or next door, or division of plant industry. So we are a team of 4 people and 2 undergraduates that are paid by our colleagues or in the Department of Entomology and Morphology, Dr. Marbrana. We have Nick and DJ, and also we have 3 dear colleagues that they are research associates that are working with the field, as Dr. Erick, Sel Smith, work with Affiliate, and Dr. Bill Gruner, working with Pentatomoidea. So before I start with the journey of my fieldwork, I would like to introduce very brief introduction of my colleagues that thanks to them, we have been working in an integrative taxonomy and systematic of the genus Anastrepa. So you can see here, we have colleagues from USDA, DPI, NC State, and UF, mainly to develop some techniques, molecular techniques, and also using morphology to advance the diagnostic tool of Anastrepa. This is for adults and also for larvae. So this is the specific goal that all the Anastrepa team share. We, I mentioned, we are using different molecular markers, and also anchor hybrid enrichment to produce a new, more robust phylogeny of the genus. And also trying to find another molecular markers for diagnostic. And the ultimate goal will be to find some markers that we can identify, for example, larvae at the port of entry of the US in a few hours. That's one of the goals. So also, I cannot leave our foreign collaborators, because without them, it will be impossible to have fresh material of this adult and larvae. As you can see here, we have colleagues from different countries, colleagues that work in different museums in Central and South America. We have people that work for the Ministry of Agriculture. They have been very helpful to go some places and to provide some logistics in order to collect these plants. Of course, I will acknowledge the very good support from the Division of Plant Industry, UAF, and also, of course, the USDA, who have been providing the grants to be able to go to these different places. So this is what I'm gonna be talking today. I'm gonna describe a little bit of the genus, provide some information, and some of the methodology that we use in the field to collect these lice. And I will spend more time on the research facilities and the logistics in order to undertake this project. At the end, I will provide a brief result, but I hope I have So the genus Anastropha, it is occur only in the neotropics, occur from the southern US to northern Argentina, but also occur in the Caribbean island. Today, this genus have at least 329 species. And of course, as other insects or group of insects, this genus has some pet species. And that's one of the reasons why we have been working with this project, Anastropha project. You can see here there are some species. One of these species occur in Florida. And one of the objectives of this project is to avoid any introduction of some invasive species like Mexican fruit fly, West Indian fruit fly, that it is in the Caribbean. And we have been lucky that hasn't come yet to Florida, but it is near, is in the Caribbean and the Gray Antilles, Cuba, Jamaica, and the other islands. So, these are the places where we have been working, mainly in the Western Amazon, and you can see here, at least in 3 different countries: Ecuador, Peru, and Bolivia. Also, we have been working the other part of the Amazon, like Suriname and French Guiana, Panama, and Central America. So we have used 2 different methods to collect these flies. One is to collect the adults. We use multi-lure traps with two component lures to attract the adults. And so far we have been very successful using this combination trap lures to retain the flies and preserve the specimen in good condition for DNA. We have using 100% provenance. And the second method we have been using is to walk through the different trails that these different research stations have and search for fruit, mainly infected fruit. And when we find infected fruit, we have to document everything. We have to see if the fruit have the larvae is feeding on the seed or the larvae is feeding on the pulp of the fruit. We need to mark the trees. We have to, in many cases, we need to hire people to climb the trees and get samples from 10 to 30 meters high in order to get a nice samples to then, get in contact with botanists and get a very good identification of the host plant, because this is very important. This is very important, the host plant species.
Oh, sorry.
This is what I would like to walk you through, this journey to some places that are in the Amazon forest. So, so with these slides, I just want to show you or tell you one experience that it couldn't happen only to me. You can see here these 4 guys, they're talking about, oh wow, we saw a jaguar. But I was the only one that I couldn't see it. And you know why? Because I was picking up fruit from the ground. I was serving the fruit and say, oh, I have mine or not. But and this guy, they were ahead of me and say, they were jumping and say, I was trying to figure out what's going on there. What happened, guys? You didn't see it? What? The jaguar was here. And then I told him, I'm sorry, I was collecting fruit, But at least I was close, but but this experience, this day, I will not forget because this happened on Sunday. And I went to the field with this guy. We say, we'll take a walk for a few hours. Do you want to come with us? You can come along. And say, yeah, I will go. I always interested in finding fruit. So we went. So this day he helped me to figure out what happened at least a week or 2 weeks before that. I was walking by myself in other different trail there, and I heard the same sound, like when this big cat jumped, like you can hear something like some big very loud sound. And then when that happened, I heard the same and start thinking, oh, I think it was the jaguar that was, when I was passing through some area there, I passed through there, and then when I passed from some place there, I heard the big sound. I think the jaguar. Yeah, I scared me, the jaguar. So I figured out that might happen before. So you can see here, this is a very nice place. After you walk for 3 or 5 hours, you can go to at this waterfall. You can swim. It's very refreshing. It's a very cool place to be in there. So in this same research station, this Villa Carmen biological station, you can see the scene. You have primary forest, we have secondary forest. They have at least two different habitats. They have river floodplains and also steep ridges. One thing I can mention here is that in the steep ridges, like in this area here is where we have been collecting more flies. Here you can see the Piñi River. So the other good thing here is that you can rent a boat. You can go upstream the Piñi River and you can visit other areas they have, because this station has 1,000 acres in the Amazon. Also, you can camping, or they also have they have host some mountain bike competition that place. So just talking about food, if you know the Peruvian food, they provide the 3 different types. They have the coast, the Andean, and also they have the Amazonian type of food. This is very good, very good. So just to mention here, this is Luz Ruiz. She was the chef. She passed away a week ago. Unfortunately, yeah, I got the bad news. She was very good chef, but also she enjoyed to go with me to different trail and look for fruit. She found at least 2 different fruit for me. She was very good. So this place have a nice lab space. They provide storage for long-term projects. They have refrigerator if you want to store sample there. It's a nice, neat place. The transportation is part of the logistic of the little scare, because you have to take a car like this one from Cusco down to Villa Carmen. You can see here, there is a lot of cliff. When you go through that road, you have a lot of cliff like this. That's very consistent from Cusco to that place. But it's a nice scene, but it's little dangerous. That's right. Okay, this is this other research station. Sasicra, this is more deep in the Amazon rainforest. These have more pristine primary forest. The elevation you can see here is lower, but still have very nice diversity of these flies. So here I was lucky to see a puma. I didn't see the jaguar, right? I saw a puma and tapir, peccaries. I couldn't see the anaconda, but it is in there. So they have some lab space, good lab space. So this is part of the transportation and logistics. You can see here, here's the bit brief while we were in the boat. So this is a third place where I stay. This is very nice. This is the northeastern Peru. This is a nice place. If somebody like to do some research in the canopy, they have a canopy walkway there. It's a very, very cool place. This is the lodging. Yeah, that's type of the lab setup. And the good thing here, in order to get to this station, you have to go through the Amazon River. That was very cool for me. Okay. So, so in order to get to that station, you have to take the Amazon River and then the Napo River also. You have to take the nice thing there is you can see also the pink dolphins in the Amazon and manatees. This is one of the headache. I will have time to just mention. So, yeah, please. It's about the permit. Today is easier, but the beginning of this project in 2012, it was a headache. It took years, 1 year, 2 years. At the beginning, they're required to apply for a research permit, another application for genetic, another application for export permit. Now, if you apply for research permit, you are covered for genetic. So you need when you apply and you include in your proposal that you will be doing morphology and also molecular, you get the permit. Then you have to submit another application for export permit when you need to retrieve the specimen to the US. So that's what I would like to have. I have more results, but I'm sure we will have the opportunity to share with you more of this. Thank you.
Thank you, Eric. Amazing talk, amazing pictures, and beautiful places. Do you have any questions for Eric? Do we have questions on Zoom?
I'd like to make a comment.
Yes, of course.
Eric has kept the bycatch from some of these fly traps and included it in some amazing Pentatomoidea. I described the new genus and species based on this material. That's nice. More people would keep all their bycatch.
Okay.
That's a good point. Also, Mike Thomas got some good stuff too. Something I can describe some.
Thank you, Eric. Thank you again. So the next speaker will be me. Okay.
I'm not ready yet. Still gotta do screen share.
Okay. Okay, so I'm going to present the distribution of an exotic ground beetle in Florida and talking about citizen science records. So you probably understood from a few talks that we had this morning that Florida is a gateway into the US for exotic species, and for different reasons. We have 12 international airports in Florida and 14 ports. There are more than 120 million visitors every year coming into Florida. And all this movement of people and goods coming from the Caribbean and Latin America, of course, increase the opportunity for exotic species to enter. And here they found good food and subtropical climate, and so they can adapt and get established. And we now have around 10 species per year that arrive and establish in Florida. So how do they travel? There are different ways for exotic species to travel and, of course, move outside their native country. And this is a list from an article from 2017. The list is longer. This is just one part of the list. And that is on the right, you see the interception that we had from 2007 to 2015 in Florida. There were intercepted around 230 species. 4% of them were found in packaging material, basically 24% in travelers' luggage, and 72% in cargos, so ships and airplanes. So today I'm going to present you this tiny ground beetle, Tetragonuderus clavigatus. He is native from South America. The type locality is Montevideo in Uruguay, and it was first recorded in Miami in 2007. And it probably was introduced accidentally through commerce, and subsequently the adults move through flight, possibly. And so this is this tiny ground beetle, and it can be recognized by having 4 spots on the elytra. The point is that this beetle belongs to an assemblage called Quadriguttatus assemblage. And so there are other 3 species similar to that. You can see those highlighted in red are the features that you have to look at to distinguish the Levigatus from the other 3. The spots on the elytra is not a really good character because it can change also within the same species. You should look at the pattern that you see on the elytra, but sometimes it's very difficult to look at that pattern if you don't have good magnification and good light. The best way to go is to extract the genitalia. And so that is a pretty good character. So keep in mind how difficult it is to distinguish this species from the other 3 that belongs to this assemblage. So I want to present you the distribution of these species in Florida. I use different data sources, insect collections, the back closet, so the UCFC, the Florida State Collection, the Archibald Biological State Collection, and a few personal collection, basically Skyle material. And also I wanted to compare these data with those that you find on GBIF. Basically, there are data from the NEON project and from 2 citizen science platforms, iNaturalist and BugGuide. Just to give you a few information on the citizen science platforms, BugGuide has right now around 500,000 observations, and it uses a more conservative approach. It means that users have a limited action on BugGuide, and there are a few editors that can make changes to the site. iNaturalist is less conservative, and you can see also how many observations we have now on iNaturalist, millions. And when you upload, of course, probably you already know, but when you upload a picture on iNaturalist, the artificial intelligence system can suggest you an identification. BugGuide, of course, does not have this system. And any user can submit a taxon identification on iNaturalist. And there are staff that can moderate the site, but they are not defined as, of course, expert. They don't have the expert status. So basically, anyone can submit identification on iNaturalist. I want to give you an example of the use of observational data in publications. This is a paper from 2017, and the authors use this species that is native to Japan, this leaf hopper. It was recorded in North America in 2005. And in this paper, they mention a new state record for Massachusetts using the photo voucher from BugGuide, and they give let's say the identifier number of the picture. So we have the photo voucher for data that we use from BugGuide and iNaturalist. And iNaturalist goes a little bit further because there is this research grade. It means that when you upload an observation on iNaturalist, of course, this observation has to include the date, the spatial or georeference, and of course, we need a picture and a sound. So the observation at that point can be qualified for the research grade. But we have to have a minimum of 2 out of 3 agreeing IDs on these observation to get to the research grade level. And iNaturalist says that you can use these observation for scientific purposes. Right now, there are more than 55 million observations that have this research grade. These are some of the pictures that I found on iNaturalist of the Tetragonoderus levigatus, because I wanted to see if there was something on iNaturalist about this species. And actually, I don't know, I'm a little bit confused because we know that the species is present in Florida. But actually, we should look at some features to be sure that this is really levigatus in order to be sure. So we should look at the pattern that we have on the elytra, and also sometimes it's not even enough. We should have the genitalia to be sure. So I don't know, probably there is a lot of guessing. I'm not saying that these are wrong, but still, probably we have to be careful. And that's why I think that the research grade sometimes can give a false sense of quality, because we can have a few identifications that are wrong on iNaturalist, and users can confirm the mistakes done by others before them. Some of the users are eager to become the top identifiers, so maybe they can give a name and that's not the right one. And also I found out that there is this Cunningham's law that some users apply. It means that users, in order to get an answer from an expert, they tend to post the wrong identification in order to, like a bait, in order to get attention from the expert. So I decided to divide the occurrences from those data that I collected from insect collections and those from human observations. One interesting fact is that I found a specimen at the FSCA that is from 2004. This means that this specimen precedes what we knew about this species, the introduction of the species, that it was 2007. So in 2004, it was already present in Florida. And we have, of course, neon. Those are the 2 sites that neon has in Florida, and those are the data from neon. And just one in Sarasota that is from another project from 2011. And as you can see from BugGuide, we only have 2 records, but from iNaturalist there are a lot. So it should be good to have so many records in order to amplify our understanding of the species and the distribution of the species. So right now, the distribution in Florida is we have 9 counties that we are sure we have specimens or DNA in collections, but we have also 6 counties that comes from human observations. So that would be good to confirm. Actually, Florida, of course, you can see that some of the counties are close to each other, so that's pretty likely that the species is present. So we only need to confirm that this species is present in those counties, and so we can confirm that. And also in iNaturalist, we can find also new state records. There is an observation from Georgia, one from Louisiana, and there is one from Puerto Rico. I'm dubious about Puerto Rico because, as you can see from the paper from 2008, that is the paper for the first record in Florida. So they gave also a distribution of the other species of the assemblage. You can see that in Jamaica and also Dominican Republic, there is the other species, Quadriductatus, and they are very, very similar. The only way to distinguish them is to look at on the elytra, the pattern on the elytra, and of course the genitalia. And from a picture on iNaturalist, a lot of them, as you could see, they are more all in movement. some of them are really tiny and the specimen is really far, so it's difficult to look at these features on a photo. And so I'm not sure about the Puerto Rican occurrence. So just a discussion on citizen science records. I would say that it's open, it's an open discussion. There are a few problems. We don't have a voucher specimens to, of course, go and look and confirm the identification. Sometimes we have poor quality of photos, and the AI system is not always right. And there are users that validate mistakes made by others. And the research grade, in my opinion, is still an imperfect process, because sometimes the observations get to this level, but it's not really true. But there is a bright side. Of course, there are species that are very easy to identify with a picture. And of course, when we are sure that the species is really that one, we could lead to an increase in species detection. So most importantly, for exotic species, so we can increase the detection of these species. And we can also increase the spatio-temporal coverage for monitoring. So when we know that a species is present in a state, we can use those data from this citizen science platform to increase our understanding of the distribution of these species. And just to conclude about the Tetragonoderos levigatus, now we know that the date of introduction is 2004, not 2007 anymore. It's well established in Florida and is gradually expanding its range. So we see that it's going to Central and North Florida. Probably it's also expanding outside the Florida. So like in Georgia and Louisiana, probably we have observations there, and also an expansion in the Caribbean region. And with this, I want to thank a few people who have me collecting records and also specimens in the field. And I thank you for the attention. Any questions?
No question.
Yes. Oops, I can't.
So, this is not a pest, right?
No.
So what's the impact of this insect, exotic insect versus that exotic pest?
Okay, a pest, it can create damage to native species, ecosystem in general. But this is considered exotic because it comes from outside, but we don't know if it is a pest. Right now, it's not. But you never know. Of course, you have to monitor and to see if it can create damage to something or someone. Yes.
Okay. Yeah, because I was thinking that probably it's exotic, but he actually has an impact in the local fauna, right? So,
yeah, it could be. Yes, we don't know. And the ecology, we don't really know a lot about the ecology of the species. There are a few papers from Brazil. And they say that probably they are seed eater, they are very abundant in soybean, cotton fields, but probably they can be also generalists. So, because I found these specimens from Orange County and Osceola County in pollinator gardens, so native plants. So probably they adapt very well here. Okay, any other question? We have time left, no? Okay. So I'm gonna call the next speaker is Felipe, and it starts his synopsis of the species of Troglophisa with 0-5 eyes. Please listen here. Oh, there.
No?
Yes, we're ready.
Well, thank you very much. I'm going to talk about an exotic springtail. So the genus Troglophisa belongs in the family Pyronellidae. They're very similar to entomobreeds, but they have a few distinguishing characters. Early last year, my junior co-author Wynn sent me some Troglophisa from a cave in Belize for identification. I had worked on this before. I described, in fact, 2 species from caves in Belize. And for that paper, I even published a checklist of the species. I even had a map with the distribution and everything. But guess what? I didn't organize that data. So I got these 2 new forms and I have to do it all over again. Okay, so I decided that this time I was going to formalize all this information so I don't have to go chasing rabbits again. So the goal of this particular work is to, was to build an annotated catalog of all the species, identify the morphological characters that are most informative for the group, and to summarize all of this information, do succinct descriptions that can help me in the future. Now, I'm going to do this. This part is only for species that have 0 to 5 eyes. So the first part, it was fairly easy. I had the papers, I just had to find them. As all of us taxonomists know that a lot of the literature is scattered all over the place and it's difficult to find sometimes. That I had from the previous work. So I have a preliminary Excel file with the information. And the result of this part was that I found 84 combinations for the species. They come from, so I had to look 5 different genera. The species were scattered in different genera, showing that there were problems with the classification. But there's only one questionable synonym, which to me that also indicates that people are not really working, because when you're working a lot on a group, you get a lot of synonyms. Many people working independently. It turns out that the genus is restricted to Africa and the Americas. And this is the general distribution of the 83 valid species of Troglodytes. In the Americans, they go from around here in Mexico to northern Argentina. And they are in tropical sub-Saharan Africa and in Madagascar. The African fauna, well, it's not very well known. There are 13 described species. 11 of them were described before 1970. They were published, many of them were from German colonies. So you can imagine it was a long time ago. And what Davide was talking about, terminology, the description from 1913 in German don't resemble anything from current German descriptions. The terminology is different. I couldn't figure out what they were. I had to ask for help. This is a species described from Ivory Coast. Most of them were described from French colonies. And this one is the last one to be described. I described this one from Madagascar. Now, from the New World, there are about 70 species. The dots here mark not the number of species, but they represent the country where they are found. They're in Mexico, throughout Central America, the Antilles, Northern South America, Brazil, and Argentina. And I have 2 unpublished records, one from Florida. There is a record from the Keys of a species described from Puerto Rico. And I have a bunch of collections from Panama that I still have to describe, but the species has never been reported. The genus have never been reported from there. Going back, so 38 of the 70 species are surface forms, and they have 6 from 8 ocelli. And this is more or less, many of the surface forms look like this. They're very colorful. These forms here are from Brazil. That's from the Dominican Republic. This is from Martinique. That's also from Martinique. That's undescribed. And this is from Panama. But many of the surface form have all these interesting color patterns, with many of them have these 2 spots in the back that they make in different ways. And to me, they look like eyes. So 32 of the 70 species have 0 to 5 ocelli. Most of those species come from caves. There's, I think there's only one species, surface species, that has fewer than 6 to 8 eyes. Most of the species so far described are from Brazil, in part because there is a colebologist, that is a biologist that helps some of the mining companies. And in Brazil, you have to make an inventory of the caves before you can mine. And so he gets to collect all these things in these mines. And they describe 15 species from caves in this genus and 100 from a different genus, all of caves formed. So the second country is Mexico, then Belize, including the 2 species that I'm going to describe. There is 1 species in Cuba, 1 in Haiti, and 1 in the Dominican Republic. Those are, the blue ones are species with reduced size number. And with these species, they are all white. Once you see one, you have seen them all. You know, they don't have the same variation in color. However, this is what they look like in alcohol. They're white. This is the species in the field. They're covered with scales, so they look completely different when they're alive. This is another of the species from Belize that I described. This one lost all the scales. That's what it looks like that. This was ready to molt, but alas, it didn't do it because that's a holotype of the species, so it's on a slide somewhere. And this is the surface species that has only 3 eyes, Trioculatus. Now, the characters used to distinguish species traditionally have been the number of eyes. There are species with 0, 1, 2, 3, 4, 5, 6, or 8. There are no species with 7 eyes. And I have 6 and 8 together because some of the species that have 6 eyes, 2 of the eyes are so small that when you make a slide, they disappear. They're there if you do a scanning electron micrograph, but on the compound microscope, you cannot see them. The other character is the shape of the hind leg claw. As you can see, there is a great variety of shapes, and presumably all of these are adaptations to living in caves. And the other character was the tip of the furcula, the jumping organ called the mucro, and may have 3, 4, or 5 teeth. So most of the species of Troglodytes described from caves are described only with these characters. Now, in 1988, my master's advisor published a paper on the paranelids of Puerto Rico. He was the first one to ever publish drawings of the chetotaxi that were detailed enough that you could actually use the chetotaxi to distinguish species. This, in fact, opened a whole new set of characters to study. So now, in addition to the eyes, the claw, and the mucro, we have chetotaxi. Those are chetotaxi of the head, of the trunk, and some of the mouth parts also have different kinds of hairs. To the point that I got 33 character systems now. And so when I atomize these character systems, I can get up to 100 actual characters. But I wasn't going to deal with all of those characters right away. Remember that what I wanted to do was to get the minimum, basically the minimum number of characters that allow me to identify the largest number of species. And so I built a matrix with 34 species. I don't have all the species yet. In fact, most of the species don't have chetotaxi. But for the species that have chetotaxi, I built this matrix. I have 34 species. I think I have to add probably 10 more with 51 characters. And the interesting thing that I found is that the combination of the dorsal macroceta and the posterior seta. See? So these are the posterior seta. So the combination of the microchaeta on the head, the metathorax, and the foreabdominal segment can distinguish basically all the species for which we have information. So the other characters are supplementary. The claw and the labium and other characters just help with the determination. But if you want to do a quick identification of something, you have to have the dorsal chaetotax. So in terms of summarizing the descriptions, so I don't have to do it over and over again. I chose a few characters. This is a comparison. These two species were described by Palacios, and they look the same way in the original description. They were both this short. For this one, I was able to get some of the types, so I was able to expand the description. But what I'm doing in these descriptions, I standardize the character, the sequence, and if the character hasn't been described, I say so. I don't leave it blank, because if I leave it blank, I can think, oh, maybe I missed that paper or something happened, I didn't get to it. But this way I know exactly that this is the next species I have to go to work on. I also did the remarks very specific so that I know the combination of characters that identify that particular species. And this is an example of when I get my hands on all those species, what we can do. Longer diagnosis and descriptions. I also put together 3 tables, diagnostic tables, and I base the tables on the number of teeth in the claw. That's just convenience, but it allows me to organize the data. And finally, I wrote a key to all the species. I redrew all the claws for all the species to make it uniform. So the key has reference to these figures. Numbers are easy to distinguish. You can distinguish easily in a couplet 4 from 5, but the shape of the unguiculus, what is that? You can describe it, but it's better to have the picture. So I have all of that also in the key. So to conclude, there are 83 species worldwide right now. 32 of them have 0 to 5 eyes. They are all restricted to the Americas. There are no African species with reduced number of eyes, probably because no one has looked. I identified 33 character systems with about 100 derived characters. All species can be diagnosed by dorsal microchaeta. There are 15 species with reduced number of eyes that we have no chaetotax information. I standardize the descriptions. These are for diagnostic purposes. So when I'm going to do a formal description, I will include the 100 or so characters, but to help with the diagnosis. And I have the keys and for all species with 0 to 5 eyes. And finally, I want to thank Palacios because he gave me some specimens to check. Clarence and Mariluz Goodnight, because they went to Mexico in the 50s and they retained the springtails. They didn't throw them away. They were collecting daddy-long legs, but they kept the springtails. And a lot of this work was based on that. Marimut, because he opened my eyes to the beautiful springtails, and DPI, because it allows me to work with things that many people don't even know exist. Thank you. Thank you.
Any question for Felipe?
I have a question, Felipe.
Yeah.
Do the species in Africa also goes into caves or not?
They all have been collected from deep litter. They were the father of soil biology, Claude de la Mar de Vaudeville, working in Ivory Coast, and he was looking at leaf litter on the floor and on the trees, and basically all the species were described doing those kind of stories. Yeah.
Thank you.
Thank you again, Felipe. Next speaker, is Eric? We don't have a James? Okay. James is presenting diversity of Taxonomic, Lepidoptera primidae. The stage is yours. And the pointer you can take.
Yeah, ready.
Okay, thank you, David. Thank you very much. Again, Eric mentioned earlier that his talk was about something that he's been working on for the past 10 years. I'm going to talk about something I've been talking about for the past 10 years. So let's see. First, I want to acknowledge a bunch of people, the curators and collections managers of some collections like Alessandra Pandolfi at the Stuttgart, almost Lice at the U.S. National Museum, and staff at the Australian National Collection at Carnegie for loaning specimens. Joelle Minet for reinstating the Lepidoptera, doing the dirty work of that difficult task. John Rollins for the postdoc opportunity to let me play around with moths 12 years ago and make some discoveries. And particularly Theo Leger and Anna Müller. Anna is an undergraduate in Berlin, and she's going to describe a bunch of species, but the question is, in what genera do they belong? So part of this purpose is to actually diagnose the genera so that you can place the species in the right genus. Lepidoptera are little gray moths. They're very diverse. Most of the species are in the genus Sufetula up here. But then there's a bunch of big, weird things that are found on old world mountain tops in the Paleotropics, Madagascar, East Asia, the Congo. And some of them are described and some of them are not described. And they're very rare in collections. And this is kind of, some of them have been or need to be transferred into the subfamily. The Lepidoptera, the current membership includes these genera. These have been transferred into it so far. As you said, most of the diversity is in Sufetula, these little gray things. I first recognized the unity of the group back in 2010 when I was reading this about an account of the Ichneumonoidea, a different subfamily in Asia, and I recognized that a couple of these things don't belong. They don't have the apomorphies of Ichneumonoidea, but they did have genitalia pretty simple. Very simplified genitalia. It was the very simplicity, the very lack of structures on them was what kind of tipped me off. It's like, these are not just simple, but they're really simple in a certain way. And I connected them to something in Puerto Rico, which is a historical pest of sugarcane. Crappy illustrations, but the student, Sain, was working under a superior morphologist, Forbes. So the morphology actually is totally accurate. In particular, they have this four-ring CUP vein. I picked up on that. Also the lack of certain structures on the male genitalia. And another interesting thing is the simplicity of the abdominal tympanal organs. Joe will talk later, I think, about noctuoid tympanal organs. The organs, these are the ears on the abdomen of moths, effectively, and they're a bit simpler than they are in noctuoids. So don't be frightened by any of this stuff. Sufetula includes a lot of species. Many of them are economically important. There's a long history of infesting the roots of palms and coconut palms and things like that in the various tropical countries. More recently, they've been getting into the ornamental palm trade. You go to a box store in the eastern US or anywhere in Europe and buy it, bring it home, IKEA, Lidl, and a moth will pop out. Very likely it might be an undescribed species. This is what the caterpillars look like. They nibble in the roots of palms, tunnel in like that. New species are being described. 2 years ago, we described a new one that was infesting pineapple roots in big plantations in Costa Rica. It's not just palms, probably other kinds of monocots, evidently. This one was described from a single specimen that came out of a potted palm earlier this last year. I do not recommend describing single things from single specimens that your friend buys from the box store, but it's In this case, they're probably getting, Nell is probably gonna get away with it because there's so many undescribed species that just by probability, yeah, okay. Anyway, so what happened was that Joel Minet in 2015, who's one of the best living Lepidoptera morphologists alive, and he had sunk this group back in 30 years before, but on reconsideration of a number of papers, he reinstated it. One of them I published was a description of a new species in Florida. There's 2 in Florida. But there's other bunch of stuff that's come to a head. He compared them to 2 other families, subfamilies, Accentripinae and Middleinae. I'll come back to that at the end. So, okay, the constituent members so far include Laphrotelias obscura. This is the only genus that brings along with it a family level name, the Laphrotelidae, therefore Laphroteliinae, one species from the South Pacific. This species, Diplocystis, has been bopping around. It's from Asia, or okay, the genitalia match stuff from New Zealand. This could be undescribed, but anyway, it's got into Eastern Europe, and I mean into Europe, Western Europe, and it's been bopping around. There are a couple of other things that have been described. More things that have been added in subsequent years, this and that, and eventually I realized that there's a few other things that I think should be transferred into it. And so now's the time I've got to publish on this stuff before somebody else scoops me on it. Lichia is actually, Minet mentioned it, but his account was more of a morphological discussion than a taxonomic one. I've got to recharacterize the Lichia. Bonipa from the Himalayas, this big brown thing. Neobonipa from the Andes, and a couple of other little gray things. I'll get into. This is what they look like. Again, many other from type specimens. John Heppner has collected large numbers of this thing, though. A lot were good with specimens for that. And there are some things that look like it from their descriptions, but I need to get specimens. Okay, so how did we actually get to this point? The thing is that many of these big, weird things have been misplaced in other subfamilies for 120 years, because the 120-year-old classification, that we have has problems. Chenobioinae was historically based upon the absence of a tongue. Well, the tongue, the proboscis, is easy to lose in root boring things. So half the stuff many things have been pulled out of Hampton's Chenobioinae. Likewise, a couple other subfamilies are based on wing venation, particular stalking patterns of the flooring radial veins. And these are no good any longer. The thing is that despite their more homogeneity of other morphological characters, the radial venation system up here, up in the costa, goes haywire in this group. There's all kinds of different stalking patterns that led them to be placed in these other subfamilies historically. And these, okay, so you can see from all of these accounts that I spent a lot of time on this. This is a taxonomic history of this thing was described in Prioraustinae, and it moved to Intercontinental, back to this thing, and then moved into this, yada, yada, yada. Okay, so we're gonna clean this up. And then, okay, so for the past 10 or 12 years, I've been running around babbling about, I've got these big weird things like that from their rare collections and so forth. Oh, they're new genera. Oh boy, yeah, right. Well, okay, maybe they are, maybe they aren't. I love their wing patterns, they're pretty. And some of them actually have, if you actually sit down and make wing slides of them, they do, okay, this thing is a new Neobonipa. One specimen from Montaigne, Colombia that Steve Pessoa gave to me. It's closely related to the one described from Peru. That makes me excited. I'm less sure about these things, but it depends on how you code stalking patterns, okay? The fact that it comes down to characters coding. So yeah, this is the story I've been talking about for the past 12 years, okay? It's kind of like, okay, we've got these, this ancient primordial stock of this ancestral lineage, and we've got these rare things coming out like Acropentius, and they're relictual, and they're only in mountaintops and Gondwana, and then out of that, and they're almost extinct, but out of that emerges this radiation of little gray things. And they're hyper diverse, and there's all kinds of applied applications, and they're economically important. And they're in your backyard too. So sort of secondary radiation in this. So this is my this is why I keep crayons in my office. Well, it's time to actually sit down and code these things and test this scenario. So the first thing you have to do is to find a sister group to this to root the phylogeny. So this is actually a few of these little gray things have been actually included in Théo Leger a few years ago published this tree. And the sister group turns out to be a subfamily called the Musotominae. Okay. Musotomines feed in ferns. They're foliage feeders in ferns. Okay, well, that's interesting. They're known to some of us because some of these Asian things have been tested for many years in the FBCL for old world climbing fern. These are vouchers from Susan Wright that she came out of her office when she retired. The morphology kind of makes sense in some way. Lactivated catosemaata are absent. The forewing is kind of has a sort of incised in this way, sort of, okay, maybe. Well, there's almost no branch support for this. This is Bayesian support. Well, yeah, 100% Bayesian support. Woohoo. Oh, we know Bayesian now. Support values are always inflated. There's no likelihood of support for this. Okay. So what I did was I started to score a data set. And I started with like a Musotomine. And I started with 2 Musotomines, thinking, oh, this will be easy. And before long, I realized that I would have to score out groups of like half of the family. So like about half, the good news is that, yeah, okay, it's the subfamily Lepidoptera. This yellow here is it's clearly monophyletic. They've got some unique characters. Musotominae comes out here. But we have a couple other things here. This is where the Middleinae and Centropinae turn out. Pretty close to it. Low support values. These are Bremer supports. But, well, the little gray ones are here. And most of the big odd ones are actually a derived in-group. Yes. But then we've got this thing from Madagascar that was included. Okay, that's down there. I've only coded that from the literature. I need to get a specimen or consult BNA about it because he's the one who critically looked at that. Part of it is because, unfortunately, I love genitalia a lot, but there's not really a lot of variation in the genitalia. There's not much information, surprisingly. This is the Centropinae, a typical Petrophila species. The caterpillars of these are aquatic. They feed on a variety of aquatic weeds. This is Hosatia, a middle line. It's a greasy specimen, but they tend to be greasy because the life histories that we know, they bore into Araceae, into Arums, also the monocots. This beautiful specimen of Middle Lamia was collected actually by, I think, Joe and you and Jim Wampus and Roy Morris. Your name's on the label for this thing for French Guiana. So thank you. This is a beautiful thing. This is another middle line. They're giant. They look like Saturniids. They're all neotropical. So I'm just going to flash you a few more trees briefly. This is preliminary. These are really preliminary analyses, and I'm not publishable really. If you pull out various taxa and add some of the described things, you get slightly different results or seriously different results. In this case, sometimes the big things come out as a comb at the base, and the little gray ones this is more in line with the narrative I was talking about earlier. But with this highly pectinate backbone here, I really suspect it's just an error, erroneous. So I'm just going to talk about this tree right now. We've got this undescribed thing from Madagascar and Diplopseustoides down here, but most of the big things are down here, up here, derived in group. So, okay, at least this gets to us as to a point where we're able to provide diagnoses for the taxa. So the Laptotelines have a bunch of synapomorphies, like some are genitalic, but they apply to all the species. Others have to do with the loss of Ketosemata. Supetula is one question. Okay, how do you distinguish this from a bunch of other little gray things? I've been thinking that all the little gray things should be synonymized together in one Supetula. Well, actually, some of them are distinct. One of these things, another thing, the big large body genera have a few of their own characters, like the number of frenulum bristles, sclerotization of the ductus bursae in the females. One character I've been paying attention to a lot is, where a couple of them are these two things. The presence of this vein here, the CUP vein, is unusual. And I've always thought that, this is a plesiomorphy. It's also found in a couple other subfamilies that also have Ichneumonoidea and Centropinae. It also seem to have all diverging in for other characters, early diverging. And likewise, this Hamus, this part of the forewing. Turns out I think I'm wrong with regard to this, but I'm probably right about with regard to the other one. This seems secondarily derived, but this does seem to be primitive. Okay, I'll skip over that. The final slide I want to point out is the tympanoid organs here. These are these little bubbles here are what they hear with, but there's this flat
area behind it, which is unusually flat and shallow. It's whereas there's a transition series, it shrinks in size, but in most other crambids, it's a deep pair of invaginated pockets that stick into the abdominal cavity down here. Most other subfamilies, but in middlelines and acentripines, again, they're shallow things. They're little, like almost like tea saucers. This is like a teacup, and there's a saucer that it lays in. Okay, so this actually makes sense, and this is something that Mine picked up on in his 2015 paper. So what I'm saying is that my morphological analysis just follows up and corroborates what Mine was saying earlier. And we need to take a second look at the Ichneumonoidea pivotal organs. So finally, genera diagnosable. If it's sister to middle-line, it also makes geographic sense because the majority of these big things are in the old world, whereas the middle lines are all neotropical. But then again, many of these things are in, most of these big things are just an in-group of the little green things. And then finally, and most of these things can also be accommodated in Acropentias, which is this thing here. We don't really need new genera for a lot of these. And that's all. Okay, thanks.
Do you have any question for James? No one from over Zoom?
I don't see any comments in chat.
There's some questions.
No, I do not see any questions.
Yeah.
So our next speaker is Erick. The title of her presentation is Grass Infesting Mealybugs and the FSA Coquilla Collection. The stage is yours. And the point is. Hi everyone. So today I'm going to be talking about grass infesting mealybugs. So just to kind of start off, here on the side we have two common grass infesting mealybugs in Florida. One Stomatomeryx that just sits right on top of the leaves and puts out this crazy wax. And then Antonina graminis that kind of goes down towards the base of the grass and puts out this long shoot to get rid of the honeydew so that it's not gumming up the body down there. So there's a lot of diversity on grass. Okay, now we're moving. Across the world, there's a little bit over 2,000 species of mealybugs currently described, and about 600 of those can be found on grass, and a subset of those are actually specializing on grass. So within Florida, we have about 23 genera that will get on grass, and probably about 8 of those are specialists on grass. So they may be very conspicuous out on the leaves. They also get in the sheaths of the leaves, which is a great place for them to avoid predators and parasitoids. And they also can get down on the roots and be subterranean. As far as our inspectors go at FDEX DPI, we don't get a lot of grass submitted despite all of this diversity and abundance on grass, probably because only a couple of those species are really conspicuous and out there in the open. But generally, we just don't see a lot of it coming in, so we're relying on us to go and collect and find more of these guys. So I kind of got dropped into this project on Tridiscus when I first started at DPI. Doug Miller and Lily Dieter had collected some undescribed Tridiscus from a nursery in Newberry on Spartina grass. And they had actually kept it in colony in the greenhouse. So we had a good supply of it to have a good series of it. But it turns out that Tridiscus probably wasn't the best foray into mealybug taxonomy because it's a bit of a mess. So there's currently 8 species that are recognized in Tridiscus, and we have 6 in North America, and then 5 in North America, and then 3 in Africa. So the ones in Africa probably don't actually belong with the North American ones, but morphologically very similar, which I'll get more into. So one of these is going to get kicked into Discococcus, so we'll be down to just 7 recognized species, plenty more to describe. So as I mentioned, this first popped up in a nursery in Newberry, and then we went out to the same nursery in Newberry in 2022 and collected it again in potted Spartina grass. And the colony has yielded all these life stages, including a good amount of adult males that I finally figured out how to rear out and actually capture because they're incredibly tiny and elusive. So we have a great series of immatures and adult males and females to describe this thing, which is great because oftentimes, as I'll mention, adult male mealybugs are forgotten. So I'll talk a bit about the morphology of Tridiscus, show you some SEM images,
talk a bit about the molecular that we've done, and just mention a couple of the ecological associates that have come up by rearing this thing in colony and greenhouse. So interestingly, we had a box of Tridiscus in the FSCA collection, and we actually found two series of slides that match our new species that we found in Newberry. And these were both collected in the 1980s, one on Spartina and one on Disticlus. And luckily and shockingly, they'd actually mounted a single adult male. And adult males are incredibly rare in the collection. But this male helped us confirm that, it was the same species, because oftentimes you get mixed infestations in the grass. You're not exactly sure which male belongs to which species of female. So it was great to be able to confirm that and have additional localities for this, as well as additional hosts. So the goal of this project is to describe the adult female, the adult male, and all of the immatures. Of course, the immatures don't have as many characters, but hopefully they'll be helpful. The adults are already troublesome enough with not giving us enough characters. So for Tridiscus, basically the main character that Ferris used to determine what a Tridiscus was that they had multiple circuli. And I'll talk a bit about what circuli are. But as you can see, as I'm listing these characters, we have these exceptions. So Tridiscus sporoboli, it only has one circulus. So it doesn't really fit that character. Having one pair of poorly defined cerari, which are these special little setae that are associated with the filaments that mealybugs make, that's really common in grass infesting mealybugs. Probably not that great of a character either. I'll talk a bit about what the osteals are. They're basically like these little defensive mechanisms that mealybugs use. But a lot of the keys that have been used for these species of Tridiscus have relied on antennal segment number, which is very variable. So you have 6 or 7, and a lot of times you're looking at a specimen that has 6 on one side and 7 on the other, and they're asymmetrical. So not very helpful. And also circuli number, which is also incredibly variable. So not a lot of great characters to define Tridiscus, and then not a lot of great characters to differentiate the species that are already in Tridiscus. And then we have Trionymus, which is another grass infesting genus with over 100 species. That is a mess, which we'll see in the molecular later on. And differentiating a Tridiscus between a Trionymus is not that easy, except the Trionymus usually doesn't have more than one circulus. So what is a circulus? So if you're looking at a side-mounted mealybug, basically have these circles on the abdomen, but they actually have depth to them, and they're kind of like thought to be a suction cup that allows them to adhere down to their host. And it's possible that they could also secrete something, maybe something sticky to help them adhere to the host, but that's currently unknown. Oops, I'm going the wrong way. So first of all, our new species of Tridiscus on Spartina. Typically, most specimens have 3 circuli, and there they are. However, we have a specimen with 2, and we have a specimen with 4. So there's some variation in the circuli number, and most descriptions of the other Tridiscus species acknowledge that they have variable number of circuli, despite relying on that in the keys. So the cerari are the cerarius. These are special conical setae that are on the margin. So if you think of like a traditional mealybug, they have these filaments that come out of them, and each filament is associated with a pair of cerari. So they're kind of supporting those wax and creating that wax with the pores that are associated with those 2 setae. So most grass infesting things, they're within the sheaths. They don't really have all the cerari coming out of the sides. Typically, they'll just have one on the anal lobe or none at all. So our Tridiscus new species has these long, thin cerarian setae, and we're finding that this is also a little bit variable. And in some of the other Tridiscus, they're completely absent, but sometimes you just have these setae that you're not really sure, is it really a cerarian setae? Because they become elongated and kind of undifferentiated from the other body setae. Here's an SEM image just showing that pair of cerarian setae again. You can see wax all over the body being created by all these pores. A lot of mealybug taxonomy is relying on wax producing pores that have multiple loculi. There's these little ducts that make pores, lots of stuff going on to make a complex wax in mealybugs. So the osteals, they have a pair, most mealybugs will have a pair anteriorly and a pair posteriorly, and when disturbed, they will actually excrete a fluid out of those that deters predators. And let's see if this video is going to work. Do you have to, can you click on it? I can. I don't know. Do I do anything? We should have checked.
Yeah, sorry, I mean, I know Andrew's video was working. I'm not sure why this one isn't.
Yeah, okay. All right, so we have an anterior pair of osteals, which unfortunately Ferris decided that Tridiscus probably had no anterior osteals, and the posterior one was weakly developed and only seen on the best specimens. And looking at the undescribed Tridiscus material from the USNM and some of the actual type specimens, it seems like it's also either very hard to see or possibly variable, even though not having an osteal at all seems like it should be a really good character. So here's some SEM images of those osteals that are excreting defensive fluids. All right, so multilocular pores, they may be dorsal or ventral, and typically on the Tridiscus, they are dorsal and ventral. So these are also producing wax. And again, we have an exception, so that one African species doesn't have them on the dorsum. So again, not a great generic character. So here's some more detail of the multilocular pores. You can see in these pictures of slides, it's really hard to actually see how many loculi they have. But once we have SEM images, you can actually start to see the different loculi in those. Here's another one, and you can, so these haven't been cleaned of their wax, so you can still see the filaments that are coming out of those and easily count the loculi compared to looking at it with the compound scope. And then they also have these trilocular pores that only have 3 loculi that are on most mealybugs. So probably the best character for this new species is that they have this concentration of what we call discoidal pores or simple pores without loculi around the hind coccyx. And this can be found in some other grass infesting species, but none of the other Tridiscus have this character, and it's very rare to occur in Trionomus either. So it seems like this is probably the best character that we've found so far for this new species. Again, so you have, there's a lot of pores going on, but basically these that are just kind of have a simple ring are these simple discoidals. And then I'm going to show you some SEM images of those as well. So this is kind of interesting. I don't think there's ever been any SEMs taken of this type of pore. And because I totally failed at cleaning the wax off, it kind of gave us an interesting perspective of seeing what's actually going on with those. So it seems like they're making kind of these little round beads of wax around them, potentially could have some pheromone aspect to them, who knows? But it's kind of nice to know that, you know, they are producing wax at least, even if we don't know much else about them. Like everything on mealybugs, they're producing wax. So going on to the males. So only about 100 species of males mealybugs have been described out of over 2,000 species. So that's only about 5%. So there's been a couple taxonomists that have put a lot of work into describing male mealybugs and have made a huge effort towards that and coming up with good characters. We still just don't have enough species described to really use them across broad comparisons of the taxonomy. But like the female mealybugs, the male mealybugs only have a single claw. And you see up here, they only have a single pair of wings like a fly. And again, like Diptera, they actually have, where did that slide go? A hemiptera that hooks into the wing. Sorry, I have to go back. I thought they were in a different order. So there are no species of Tridiscus males currently described. One of the species, Tridiscus sporoboli, there is a male in the type series that was really vaguely described in the early 20th century. As basically the total body length was given, and that was the end of it. So having a redescription of that will probably be very helpful. But it seems like one of the good characters for this genus is that they have these fleshy setae on every segment except for the first one. And most male mealybugs, if they have fleshy setae, they'll be at the end of the antennae, not at the beginning. Oh, now I'm going on right here. So male mealybugs have a pair of dorsal simple eyes, a pair of ventral simple eyes, and then an ocellus. And this is not even very many eyes compared to other scale males. I think the giant mealybug Doug and I are working on has like 16 eyes. So I haven't moved far away from my background of spiders. Still keep going the wrong way. Here's the fleshy setae on those antennae, some SEM images we got recently. This was interesting. That doesn't show up on slide mounts. Not sure what's going on. We kind of have this like rugose intra-antennal segment versus a smooth one earlier on in the antennae. No idea what's going on there, but did not catch that at all on the slide mounts. So that was something that was interesting that came up with SEM. Again, that handy level tier. Sometimes they'll have multiple CD coming out of those. Most mealybugs just have one. So again, SEM has been really helpful for tiny characters. So the males are incredibly small. Here we are looking at a couple multilocular pores on the head with a 100x objective on oil with the IC. And you can see that you can't really tell how many loculi are in that. It's pretty fuzzy. It's probably going to look better with phase contrast versus the IC, but still really hard to tell. We'll get it on SEM. Clearly, it has 4. So the SEM has been helpful in that way. Oh, I had an arrow. So moving on to the molecular, really quickly, this is more Matt's side of things, so I'm not going to go into detail. But there's really been 3 major phylogenies of mealybugs done. And we have done 9 gene fragments with the new Tridiscus species. And it is coming out down here. So the only other Tridiscus that's ever been sequenced is Tridiscus distichli, and you see that they're not quite sister taxa, but we're getting closer. So there's a lot of issues with mealybug phylogeny right now. So within the Pseudococcini, there's just not a lot of resolution. So I've highlighted all the Trionomus. So Trionomus is clearly a disaster. And it seems like maybe we have convergent evolution of a lot of these morphological characters in grass infesting species. So they're all ending up looking the same. And we really need probably more molecular data to resolve these relationships. We have ants tending the mealybugs in the greenhouse, the ghost ants, and we've also had a mixed infestation with the common Antiograminous mealybug. So this is just a couple of ecological associates we found. Lots of material from Beltsville to look at from a lot of different states. Increasing molecular sampling of grass infesting things is the next goal. So thank you to the scale team, the molecular support and SEM support.
Thank you very much, Eric. Any questions for Eric?
So that little structures on the antenna that you discovered, it's different. You know the functionality?
No, it was kind of a surprise. We're looking at the SEM, so we're I'm gonna look at the slides again and see if we can actually see it now that I know that it's there. Doug, do you have any hypotheses?
See it.
Yeah. Okay.
On the slide, yeah, I looked after.
Yeah.
You can, but I have no idea what it is.
Okay. Julieta has a question. Does the Altair hook to it?
It hooks into the wing.
Oh, so it hooks.
Yeah. So that CETA hooks into there. There's like a little like.
You just the picture was separated, but okay, hook.
Yeah. So they hardly ever mount or die hooked.
Yes.
So yeah, usually the CETA is going out the way.
Yes.
Yeah. Okay. Thank you.
Thank you.
Thank you.
Okay, the next speaker will be Mike, and we have him over Zoom, right?
Yes, Mike, we can see your screen, so your screen sharing is working. You want to just make sure your audio is, I guess, please start talking so we can confirm your audio is working.
Thank you. And do I undo the video or? All right, I'm going to start. Exploring the surprising beetle diversity along I-35, running up through the center of Texas via data compiled from iNaturalist. There's a exponential increase in observational data, and the iNaturalist as even though it's 54 million research grade observations, it pales in comparison to eBird, which is 1 billion observations. So 1 billion would be like 2/3 of the total here. So if you exclude the birds here, observational data is tracking a little closer to the specimen data. And the other thing is that specimen data is going to be more species rich than the observational data.
Mike, if I could interrupt you quickly, just so you know, right now what we're seeing on our screen is your current slide as well as the next slide. Is that intentionally what you want to show us? Or
not exactly. Let me
So I have no control over your slides as you're sharing your screens, but if you can switch it into a slideshow mode or.
Well, I thought I was in the slide. I've got two screens here, so I'm. Let me close that. Now, are you seeing just one?
No, we are still seeing two. I will say though, now we're seeing one slide.
Now you're seeing one slide?
Yes.
Okay, perfect. All right. So the I loaded up the number of beetle species observed for iNaturalist for most of the states, and 4 states stood out: California, Texas, and 2 provinces in Canada, Ontario, Quebec. These are large areas, and so these high beetle numbers kind of correspond to island biogeography theory. They're also can be skewed from high population. If you note that New York has as many beetle species reported to iNaturalist as Arizona, so high population skewing that. And you can also see Monterey and Nuevo Leon is the most diverse by observational data along Mexico's northern tier of states, but Tamaulipas in reality is much more diverse. So there can be some skewing of diversity from just the number of observers in an area. And the the fact that these 2 provinces exceed all of the smaller states. So there's the diversity from iNaturalist is a little doesn't track with normal latitudinal shifts. So let's look at some actual specimen data to compare to iNaturalist data. This is Ed Riley, who my mentor since I started at A&M years ago. And so here's his species in blue, and the iNaturalist doing pretty good in Longhorn land. Bupressids, we got some Agrilis there that are missing. Staphylinids, we've got a long ways to go, but the observational data, iNaturalist, about 15 years since iNaturalist was established, and Thomas Say came through North Texas in 1820. So we got 200 years of specimen data. But overall, iNaturalist is about 44% of Ed Riley's estimation of the species diversity. These 12 families represent 70% of the Texas total. So how are species distributed? First, we generally go to ecoregions, and there's some endemism in some of these southern ecoregions, but some of the species generally do continue on to the south. But ecoregions are important, but maybe I think this is a little too fine of a scale. Temperature is generally not as much of a factor in distribution. Lots of species range from Florida to Quebec, and these are 10-degree Fahrenheit increments. So 40 above to 40 below there. One of the primary factors in species distribution is the wet areas, eastern US, eastern North America versus western North America, with returning to wet areas on the Pacific coast. But the general dividing line between east and west, the 98th meridian, the ecotone between east and west. El Paso, less than 10 inches of rain. Houston, 50 inches of rain on average. And so species that can hang in Houston have a hard time in El Paso and vice versa. So this is the I-35 is kind of shorthand for this region here, and the city populations also track similarly with these peaks in western cities are often pretty artificial, Phoenix, Las Vegas, El Paso, and such. So I-35 Duluth down to Laredo here. So the Carl Steffen, his 30 years in Latimer County, he made this one of the most thoroughly documented pieces of real estate for beetle diversity in the US. His 3,500 species is more than the diversity of birds in all of South America. So that's kind of mind-blowing there. Ed Riley did another survey of importance with 2,000 plus beetle species in Lick Creek Park, 500-acre park, just a couple miles from his backyard. So combining Lick Creek with what he's finding in his yard, 2,000 species there. So switching back to iNaturalist data, the most diverse counties that the top 5 counties are Dallas, Fort Worth, Dallas County, Tarrant County, Travis County for Austin, and Hidalgo and Cameron County in the Rio Grande Valley. Here's the iNaturalist data for Brazos County, where Ed found 2,000 plus species. And so I want to look more in depth at these 5 counties. This line here is approximately the 99th meridian, so it's a kind of arbitrary. The as I pointed out a moment ago, the kind of the demarcation between east and west is the 98th meridian, and that goes right through these high diversity counties. And this is a lot to do with the western species coming up against the eastern species. So you have a good mix of east and west right along the dividing line of the 98th meridian. So here's Latimer County, Brazos County, and with the 3,500 beetle species, 2,000 beetle species. The Fort Worth here, Dallas Fort Worth represented here, and Cameron, Rio Grande Valley, Cameron and Hidalgo counties here, and this is Austin, Travis County. And this is kind of a sweet spot in terms of soils and flora as well. The piney woods are conifer forests, and so way out here we have the Chihuahuan Desert, and this is the Great Plains coming down through these large ecoregions, and then coming through the Ozark Plateau and down through the Blackland Prairie, Post Oak Savanna, down to the Rio Grande Valley. This is temperate forest. So the high diversity in these 5 counties that have a lot of population pushing up their numbers of observers, those high counties correspond to the high specimen data from Latimer County and Brazos County, and all of this is in the temperate forests, and also the east-west meeting and rainfall median sweet spot. So a lot of factors adding into the diversity here in these Central Texas, shorthand I-35 corridor coming up through there. So looking at other taxa beyond beetles, we have vascular plants highest in Travis County. The moths, Lepidoptera, highest in Travis County. Beetles, and then the amazing parallel nature of Hemiptera, Hymenoptera, and Diptera here. So what I'm saying about beetles probably largely applies to other taxonomic groups as well. The you kind of counterintuitive or surprising is more better word, that the Rio Grande Valley, southmost Texas, has a lower diversity than Dallas Fort Worth. That's kind of crazy. Moth diversity is high because so many of them are site ideable more so than beetles and certainly Hymenoptera and Diptera. So moth photography group really helps keep the moth numbers above because we're talking about species level data here, numbers of that have identified fully to species basically. So if we look at the, there's a loose correlation between the increase in population and the red line on this scale on the right, or excuse me, left, and the species diversity. So the higher population counties, Dallas, Fort Worth, San Antonio, Austin, Rio Grande Valley, those are generally higher. El Paso is an outlier on the low end of the scale. New Mexico is currently in the worst drought in 1,200 years. So that's El Paso's tucked right up under New Mexico there. So this is interesting, and possibly as the population of the county increases, the diversity might actually decrease. But you think of don't think of Dallas Fort Worth as being a biodiversity hotspot, right? So let's look at some individual species, and the eastern. We're going to look at different species that have different life histories. So wood boring beetle hosts in virtually all eastern hardwoods, so not a host limited. It's not temperature limited. It's way up here in Winnipeg. It's moisture limited. The Calisoma species is predator, so not prey limited generally, but has a abundance of records in the I-35 corridor here. The 98th parallel really comes in here, going on up into Kansas.
Mike, I am hearing from our moderator that you have about 1 minute left of your time. We gave you a little extra to the slide mix up earlier, but thank you.
Am I done, or do I have a minute?
You have a minute.
Okay, thank you. Another eastern species, high diversity or abundance in the corridor, east-west spanning species, soil fossorial in the larval stage, 98th parallel per meridian. Species does follow its host plant here, and the Chrysomela feeds on Monarda, which is a widespread species, but it's really locked in on the the Blackland Prairie post oak savanna. There are people actually north of the Red River, but it drops off there. And then a non-native species, again, fossorial. This is the Edwards Plateau is, stone thin soils, and piney woods is acidic soil, think azaleas, and a western species way up, not temperature limited, I-35, a species that doesn't come to lights, so missed by all the iNaturalists up here. These are all trap data. And so basically diversity and abundance along I-35 corridor is underappreciated. And iNaturalist still only has about 50% of the Texas arthropod species. Thank you. So I think I'm out of time.
Thank you, Mike.
All right.
As a question, one over Zoom.
Nothing in the chat. Oh, actually, yes, we just got one question in the chat from Louis Hessler. He asks, what is diversity like in Harris County?
Harris County is where did I put that data? It's somewhat similar to 669 beetle species, so it's comes in behind Dallas County and behind the Rio Grande Valley. So this is Corpus Christi here, Bastrop County. This is the Balcones Fault Zone. This is a high diversity area, lots of people, lots of naturalists. So Houston is, I excluded it because on my graph, it would have thrown off, the population is so high, it would have thrown off the other graphs here. Right here, it would have thrown it off. So I didn't include Harris County, but it's up right in here. So this is all insect species. Thank you.
Okay, awesome. Thank you, Mike. Yeah. Okay, right now we will have the coffee break. We'll be back around 10 minutes because we are a bit late. For those who want to remain for dinner, please raise your hand so Julieta knows and she can make a reservation. Okay. Okay, perfect. So I see you in 10 minutes. Thank you.
And if any presenters have not uploaded their slide, presenters that are here in person, please bring a flash drive over to upload your slides.
Well, I had a lot to I couldn't. It's the only way to cover a lot of.
Are you?
Hi, everybody on Zoom. I think we're about to get started again. Just to confirm what both of you have probably already figured out. We're running almost exactly 15 minutes behind. So if you look at the schedule, every talk scheduled for a certain time for the rest of the session, we'll be starting 15 minutes later. I'm only saying that because if there are people you know of that are planning on joining for a specific talk, logging on to Zoom at a specific time, they will have to, join, wait 15 minutes to get to the talk.
Yes.
Okay. Welcome back, everyone. For dinner, we are encountering there at the restaurant at 6, not 5:30, because the reservation. Yes, we got a table around 6, right? Yeah. Okay. So let's invite the first speaker for this section is Han Dan. And the presentation is synopsis of the Florida species of long-tailed thrips. And the stage is yours. Okay.
So you stay here.
Hey, everybody. If we haven't met before, my name is Ann Dunn. I am working with Felipe in the Thrips et al. Lab. You may know me as the person taking up 60% of all the counter space in the beetle room right now. Sorry. And I'm really excited today to tell you about what we've been working on with regards to some of the non-pest thrips in Florida. So, you work with Ichneumonoidea, 95% of your time and energy goes into the plant pest species, but that's actually a really small proportion of the total diversity found in the order. So there's almost 6,000 described species in Ichneumonoidea, and they have a huge variety of life history strategies. So there's predatory thrips, there's social thrips that have casts and polymorphism, and there's also a huge number of fungus feeding and spore feeding thrips, which is what I'm going to focus on today. So because thrips are small and Florida is Florida, we're assuming we have a lot of adventive species that we just haven't been noticing because very few people look at these things. So as scientists, we want to keep an eye on that. And just from a fun perspective, for whatever reason, some of the most visually and morphologically distinctive thrips we know of are fungus specialists. So just as an example, all 6 of these came from one Berlese sample, so one plastic bag of leaves. And it has all this diversity of shape and color. There's winged and wingless forms of the same species. There's a lot going on. And this other guy, just for fun, this is Erachiothrips, or the peanut wing. It has these super cool inflated forewings with this reticulation pattern, almost like a lace bug if you're familiar. And no one knew anything like this existed until somebody found it. And it's a leaf litter specialist. So there's cool stuff going on that we're just not paying much attention to. So we want to know which species are native and which are adventive. How do we know that if no one studies these? We at the FSCA have perhaps one of the only opportunities to actually use that information because we have historical data on fungus thrips. No one else does that. So J.R. Watson was one of the founding members. He was the first president of the Florida Entomological Society. He did a lot of things during his career, but his love was Hymenoptera. He loved thrips. And so throughout the course of his life here, he made almost 40,000 slides, and FSCA has all of them. So a lot of them were pest thrips, but he did the non-pests too because he really liked thrips. And you really can go into the slide collection and open a box and find a slide that he's doing. So, thrips 101. If you have a thrips in front of you and you don't know what it is, you have no idea. Pretty much the first thing you're going to want to look at is the end of the abdomen, because the shape and the form of the final abdominal segment is used to separate the 2 suborders of thrips. So your typical thrips, I suppose you'd say, your Franklinella, your Terebrontia thrips, the 10th abdominal segment is the smallest, and the abdomen just sort of condenses into a point, as you would expect. But the other suborder, the Tubulifera, the final abdominal segment is extended into this structure we call a tube. It's usually about twice as long as the segment before it. So the Tubulifera There's about 8 tribes. They're kind of a mess. But one tribe in particular, the Eurythropinae or the tailed thrips, they take this idea of the tube and they really turn it up to 11. So you can see they have this really long, thin tube, several times as long as it is wide. And in addition, they have these long seedi on the end of it. They're so long and so elongated that the combination of the length of the tube and the seedi is often larger than the entire rest of the thrip's body length. So they have this immediately recognizable structure. Half of their length is tube and 2 pairs. And then just from a practical perspective, they're easy to find. So they like oak and pine litter, which we've got a lot of. Actually, a really great way to find some of them. This is natal. So a really great way to find some of them is just to take a white object like a plate or a bowl and take a pine cone off the ground and tap it and they'll fall right out. Hope this works. They're still small, even for thrips, they're small, but even with this little tiny guy, you can still see the tail sticking out. It's noticeable even in the field. This works. So what I'm going to show you here, this is like a 15-second video just for fun of behavior of some live Eurythropinae that I found, actually utilizing the tail as we think a predator defense mechanism. So what you're going to see in this video is Thrips is walking around and I come and poke it with a pine needle. And what he does is he rears up his little armored forelegs, he moves backward, and he starts really whipping that tail back and forth, like rapidly, so much so that his whole little body shakes. You think I can do it?
There we go.
Yeah, there he is. There's me. I'm bothering him. It's upset. Really like swiping that thing like really fast, really forcibly. Look at him. Little propeller. So as far as we know, that behavior has never been documented before. But that's just an aside for now. This is just one new direction we could go out of many. Okay. So based on our historical data, mostly by J.R. Watson, we think there are 3 species in this tribe native to Florida, which is not a lot. So we plan to do the whole state of Florida. We want to do all the fungus thrips. So this is our idea of like level 1. This is our easy, small, easily recognizable group that we want to start with. So we have these 3 native species. 2 of them are in the genus Trachythrips and 1 is in the genus Stephanopthrips. Basically, they're easy to differentiate between each other under a light microscope. You don't have to slide mount. Excuse me, you don't have to slide mount every single one, which is convenient. I can just pour the contents of a sample into a tray and just sort it by eye. And Watson I, yes, was named after J.R. Watson. Hood, who described all of these species, was buddies with Watson, and he would come and visit him to collect Ichneumonoidea. So the only other wrinkle that we have in this story for the past 100 years is we have one adventist species of Stephanopthrips. It was first detected in Georgia in around 2008, and then first detected in Florida by Felipe in 2017. So we have very little idea what the life history strategy of this thing is. No, almost nothing about it. But we're going to keep an eye on it. And it's also fairly easy to distinguish from the other species. So Watson dies in 1946, fortunately. The vast majority of our slides of Eurythropinae of this tribe were made by him while he was alive. So after he dies, we have almost nothing until all the way in 2017. This is when Felipe finds Japonicus. So this is a decent number of slides for a non-pest cryptic leaf litter tribe, but still, with almost the entire second half of the 20th century is empty. And as you might expect, because Watson lived and worked in Gainesville, the majority of the slides and the majority of the collections are also from North Central Florida, with like a couple scattered ones in Tallahassee and Miami. So that's what we expect. But we could do better. You know, there's huge portions of Florida that have never been sampled. Obviously, the thrips that are found in Bay Head are not going to be the same as the thrips found in Tortoise Road. So we want to check on those places. So we got this project sort of embryonic, started in June of last year. And at that point, our two objectives were to sort and identify all of the Eurythropidae we had access to in the collection. So all of the slides and all of the existing Berlese collections that Felipe has made for other things. So we do that, and then we want to find out where we don't have any samples and go there. So simple enough. So Felipe's samples are the circles. He's got a lot more than me because he's got a couple of years of doing this. But as you can see, it's about the same as the historical samples that concentrated around the north central Florida area. So we're hoping to get out of these places as fast as we can. We also have a couple samples from Jim Hayden and others. I haven't had time to update this map yet. So we've only been at this a few months, and we've already found all 4 of the species we expect, which is good. That means we're looking at the full variety of habitats that these things are known from. So we're happy about that. Now, Watsoni is the most common by far, and this also is the case with the historical slide data. Most of our slides are also Watsoni. And Seminole and Occidentalis are sort of range restricted. But Japonicus is also pretty common. We'll get to that later. So we're going through all of these thrips. We have thousands of thrips now. About 1/3 of the Trachythrips that we are collecting just don't really fit as Watsoni or Seminole. They can't be easily called one or the other. So at this point, we start to think something unusual is happening. We need to take a look at it. So what I do is I get all of the type description papers for every species of Trachythrips found in North and South America, because there's no photos or illustrations. And I read through them line by line and take every character and put it into a matrix. So this is just a piece of that. And once I have this matrix, now I can take every unknown Trachythrips we have, look at its characters and see where we can fit it. So there's a lot of stuff going on, a lot of things with the cuticle and the hypodermal pigments. But the most important character systems you want to look at are the shape of the head, the number of the antennomeres, the forelegs, and the length of the tooth. So I start doing that. I start taking all of our specimens and trying to place them somewhere into this matrix. And as it turns out, some of our unknowns are a described species of Trachythrips that's so far only ever been known from the type series, which is collected in Haiti. So we're going to get those types. We want to look at them and make sure that this is what we have. But I'm fairly confident this is good. So it's easily distinguishable from Watsoni just under a light microscope. You can see the 4 tibiae are tan and Watsoni they are brown. And also, if you put the two side by side, Brevitubus, as you may expect, has a shorter tube in general, and it's also spindle shaped, sort of. So this is cool. But that's not all of our unknowns. Not all of the unknown Trachythrips are Brevitubus. We have to keep looking. So now I'm looking at the historical slides. I'm looking at the slides that are already in the collection. And hold on, some of those also don't fit. Now, when these slides were being made in the 1920s and 30s, there was only one described species of Trachythrips. So a lot of them are just labeled Trachythrips because what else would it be? But some of these don't really fit as Watsoni or Seminole either, and they don't fit any of the other described species. So I start at this point with thinking, okay, we have something new. What I have to do is I start taking all these unknowns and putting them into groups best I can, so that we can start getting an idea of where these different forms are found. So Trachythrips are supposed to have 5 or 6 antennomeres, and this one has 7, so that's weird. So we're going to call him A for antennomeres. And the set of characters that this guy has, including the antennomeres, he's only supposed to have brown legs, but he has tan legs. So that's weird. We're going to call him L for legs. We actually have a lot of this guy. We keep finding him. And this other one I just want to point out because it's so weird. We have this one extra strange form found so far only in Volusia County. You can see its head capsule is sort of triangular. It's sort of tapering up into a point. And on top of that, its legs are also the wrong color. So there's a lot of weird stuff going on that we're just starting to appreciate. We're good. Excuse me. We're going to call him H for head. So this is supposed to be a cakewalk. This is supposed to be easy peasy. We'll pick the smallest possible group of thrips and we'll find them and it'll be great. And we've been doing it for 7 months now, and I've already doubled the number of species we're potentially looking at. So that's good, but that just illustrates how much work we have left to do, and how long this is going to take. So, yeah. Send help. So one last thing I want to mention is our adventive species, which is Stephanophyps japonicus. Now, it's pretty common. It's not as common as Watsoni, but it's pretty common. And sometimes we find it in normal numbers. We'll find, 3 or 4 in a sample. But sometimes, in some situations, we find so many japonicus that it's outnumbering not only the other native Eurythropinae, but it's just outnumbering every thrips. There's more japonicus than anything else. This is a photo of one sample, again, just over 100 thrips in it, and over 90 of those thrips were japonicus. So if you figure one of our samples is like 10 liters of leaf litter, and if you extrapolate that to an acre or to a whole forest, that's a massive number of thrips. That's a massive amount of biomass that is now this one species. So now that we're here, we have this information. Now that we know we have a difference between the past data and the present data, our next step, we're going to go back to those sites where Watson found Watsoni in the 30s, and we're going to see, do we find Watsoni there, or are we going to find japonicus? So I hope you'll watch this space. Before I go, if any of you are traveling around Florida doing any collecting ever, anywhere, further away from Alachua County, the better, and you think you can get a bag of leaf litter for us, we would absolutely love it. Please get in touch with me. My name is very easy to spell. Special shout out to Kat Daly for helping with the sorting, and Jim Hayden, Oliver Keller, and Rebecca Kruger for getting us bags of leaf litter. Please get in touch with me. Thank you guys so much. I'd love to take any questions you have.
Great presentation. Any questions for her?
What's up?
Would it be more helpful for us if we have access to a Berlin to set that up and send you that?
Yeah, if you can do it yourself, yes, that's good. That's great. I was going to say, I'll show you how to do it if you've never done it before. But yeah.
Or seen any habitat differences?
It's so early. We have some of these forms we have from only 1 or 2 places. And it's difficult for us to get to South Florida to do a lot of sampling because we live here and we have to drive. So I can't say anything yet, but hopefully we'll be able to find that out.
Yeah, it's mostly like oak leaf litter, so it's not like a host. Let's see the sheet. I've got a bunch of EMAC samples.
Yes. Yeah, that's great. Yeah, please get in touch with me. My email's very easy to spell.
We have one more question over Zoom, but I can't say it right now.
It was basically, how would you like the specimen prepared to ship, the basic prepared to ship?
Oh, yes. How would you like samples to be prepared?
Just an ethanol is fine. I mean, we're just doing Berlese sampling. So if you can do the Berlese by yourself and send us the thing of ethanol with the buds in it, you don't have to separate the thrips out because we do other things. We do isopods also. So those are good for us too.
And springtails, some evil.
Yeah. No bias.
Thank you. Next speaker, we have Brian Hermitage. Do we have him on over Zoom?
Brian is here. Yes, Brian, you should have permission to share your screen. So are you able to do that? Okay. Also, can you please unmute yourself?
All right, I'm on them. Can you hear me?
We can hear you.
All right. Now what?
So
I hit share.
All right. It's taking a minute, but it's working.
I'm pressing slideshow, but I don't have a slideshow yet. There. Are we good?
No, it is not showing up as a slideshow.
Let's try again.
Yeah. So visitor, we can also, like we talked about yesterday, you can try sharing your entire, close anything else on your desktop, share your entire desktop, and then pull up.
Yeah, that's actually what I had. But
Oh, well then I'm not sure why it's
Share screen, try it again, share.
there. I did it. Yeah, why don't you stop sharing your screen and try it a second time?
what's that?
Why don't you stop sharing your screen and then try it again, just to.
Okay. All right, so I'm hitting share screen at the bottom. And I've highlighted PowerPoint show. Hold it a second. Nope.
Well, now you're not sharing your screen at all. So if it's not working like we talked about yesterday, share your entire, close everything else on your desktop and share your entire desktop window, and then put your PowerPoint in slideshow mode. And that's what worked yesterday.
Sorry. Okay, now, right now, all I see is my desktop with PowerPoint on top.
Okay, so press share screen.
Well, I've got to get to your share screen.
And there should be an option to share your desktop. It's probably the upper leftmost. Okay, it's in the middle of doing something. So now put it in slideshow mode.
Which I just did. Do it again.
Okay,
Yep,
here we go.
that's it.
We got it?
It's working.
Sorry
right.
sorry for the bother.
Not a
Greetings
problem.
from the Western Panamanian Highlands. This is probably the only group that I can brag about the weather down here without hacking them off right now. We're going to talk about some of the caddisfly work we're doing down here in Panama. I've been doing since I arrived in 2012, 2013. My co-authors, you'll see here in a minute on the next slide. Tomas Rios on the left and Youssef Aguirre on the right. They both have Masters of Science degrees from the University of Panama. They'd never really worked in adult aquatic insects before I started working with them. This is the 5th year, I guess. And they're now to the point where they can identify species and also find new species. Now we're working on the other end, which is getting things published. We've created an aquatic vertebrate research group in 2018. And in addition to the 3 of us, we've added some additional people. Most of the increase in species has come about because of the microcaddisflies, the Heteroptera, and primary person responsible for that group is Steve Harris on your left here. We've included Robin Thompson on the right most recently. In fact, she'll be coming down here the latter half of March to do an NSF project on Heteroptera here in. But they primarily take care of the micros right now. My eyes are too old to do microcaddisflies. Roger Blanick, also at the University of Minnesota, helps with verification. Grandi Anderson and I are starting to work on the chironomids here in Panama, which I think we have a list right now in the literature of maybe 40 species, and there's probably 1,000 species, so a lot of work to do there. Kyla Castillo Sanchez is a graduate of UNACHI, the University in Cherokee, and David, and she works on stoneflies. And then finally on my right is my wife, Tatiana. She's a former senior research scientist at the Russian Academy of Science in Vladivostok. And we got married back in 2006. And a couple of years later, I said, right after she got her US citizenship, I said, let's move to Panama. And so we've been down here since 2012. And of course, she also worked on caddisflies, but now she does more of what you see in the background there, which is paint the flora and fauna of Panama. Back in 1992, Yolanda Aguila produced a paper and a book on insects of Panama on the caddis flies and summarized what was known up to that point. And there were, at that point, 13 families, 39 genera, and 168 species. And between that publication and when we got down here to Panama, an additional 6 genera and 78 species were added by a variety of authors. So that brought the total approximately 2014 up to 13 families, 45 genera, and 246 species. One of the reasons my wife and I moved down here is because after assessing this, we decided there was a lot more to know about caddis flies in Panama, and that was our favorite group, so we came. Little did we know. Here's the way things were when we came down here. In Mexico and Costa Rica, they knew a little bit over 500 species each. In Panama, 246, down in Brazil, about 750. But most of the rest of Central America, virtually nothing. Lourdes Chamorro did some work in Nicaragua to increase that country's fauna. But the rest of the countries in Central America, very little has been done. In fact, I'd be very interested in receiving material, for example, from Belize or El Salvador, where virtually nothing's known in the literature. So we began collecting, and you can see here from beginning in with the 246 in 2012, what we've added since 2015 when we first put our first publication out. And we've basically added 2 families, 11 genera, and 276 species. Actually, I've got a 12th genus, new genus, but it's not been published yet. So you can see the totals down at the bottom of about 522 species. Now I'm cheating a little bit here because we have some papers in either in press or in publication in the review mode, which should be out this first half of this year. So this goes through actually the first half of 2023. Of those new species, whether they're new species or first country records, about 1/3 of them are new species and 2/3 are first country records. We've made about 250 collections at about 80 locations. We try to do collections at each location, 1 per month, January through June, avoiding the rainy season where you don't add a lot. We prefer to do both malaise traps and UV traps in combination. And so far, in terms of publications, we put out 22 papers on Trichoptera. 12 of those were in Insectomundi. And we have some additional ones in press right now. We're in preparation. 5 species of Anacronoria have been added in the stoneflies. And we have 1 paper on the Diptera, which was sort of a review paper on the Topulidae. But right now I'm working with Tron Anderson on a couple of papers on the Chironomidae, and with lots of new species, as you might imagine. As I said, our preferred method is collecting a combination of malaise trap and ultraviolet light traps. We cheat a little bit by putting our malaise trap across the streams. We don't just put them on the side and hope that the caddis flies blunder their way into them. So we put them right in their flight path. We have 2 different types of light traps that we use. This is a pan light trap on the left during the dry season, and during the wet season, where the rains tend to come in late afternoon, early evening, we put them inside of a 3-gallon tote with a window cut on the side to keep the sometimes torrential rains out. But this combination of using malaise traps and light traps is the key, because oftentimes a given fauna, you'll get anywhere to 40 to 60% of the species that will come only to malaise traps, and the opposite, 40 to 60, will come only to the UV light traps. So they're a good complement for each other. Sweeping is almost useless. I know when I collected in Western North America, sweeping was wonderful. But down here, sweeping gets you almost nothing. It's not worth the effort. So these are the two preferred methods. Our game plan, however, involves more than just generating new species and species descriptions and first country records. First of all, we're interested in the distribution, and we've not done a really great job with that so far since we collect where we live right now. Most of our collections, as you can see here, are in Western Panama. We've done various altitudes there, but and then the other collections primarily are national parks. We were involved in a national park project for several years, and we're now branching out into other areas, but right now most of our collections have been in Western Panama. We're also interested in distribution with elevation. I'll show you a slide on that in a minute, and species packing, and trying to identify centers of endemism and associating larval and adult life stages. And we've not done a whole lot with either of those last two items, but we plan to. Here's a graph of distribution of Leucotrichia, which is a hydroptilid or microcaddisfly, showing its distribution with species. The number of records are in parentheses beside each name. Now that may look low, but these represent the unique records. For example, the first one, Leucotrichia hispida, says 4, but that may represent 24 or 30 records. I just took the unique ones from the different sites. I didn't enlarge my data by using all the different repeated records that we had. So you can see there's certain species like Leucotrichia hispida and Phthiraptera that are only found at the higher altitudes, or so far we've only found them there, and then a few on the right that we've only found at the lower. This mirrors other species and other genera, and actually another in a family that we've seen. You see the same kind of pattern where most of the species are found from lowland to midland. You have a few that are in the higher altitudes and a few only in the lower altitudes, 0 to 150 or 200 meter altitudes. So we're looking at that with a variety of different groups. On your right, this shows you some of the species packing. I guess Gauss's competitive exclusion theory doesn't hold down here with all the resources, the abundant resources we have. This is once again for Leucotrichia, a paper we published recently, showing two streams, Quebrada Rambala, which is 150 meters, and Quebrada Jaramillo, which is about 4,500 or 5,000 feet, I mean, above sea level. It'd be 1,200 meters roughly. And you see there's 7 species in each one of the same, and this is at one site, just one location. We get 7 species of the same genus. And those both, by the way, are very interesting watersheds when you consider the other species that are present too. On the left-hand column, this shows you sort of the progression for this one genus, Leucotrichia. When we came, there were 3 species known from Panama, with 2 of them described from Panama. Then once we started working, we added a new species and then 4 first country records. And then we decided to review the genus. And this was Steve Harris and Robin Thompson and I. And we added 4 more new species and 2 more first country records. So by the time we published the paper, we went from 3 up to 14 species for this one genus, which happened to be the highest diversity in the world, actually. Latin America is only known from Latin America. Unbeknownst to us, one of our reviewers forgot to tell us that he had just published a paper where he had placed, based on DNA evidence, a 15th species in Panama. So there's actually 15 species now known from Panama. So we're looking at species packing, we're looking at altitudinal distribution, and we're also following the increase in species that we get as we collect more. In 2023, we plan on doing several different projects. We've already established a malaise trap in a headwater stream, Quebrada Las Cedras, somewhat close to where we live here. We're involved in a UNESCO-sponsored geopark project, which is in several Latin American countries, from Mexico all the way down, I believe, to Uruguay or Argentina, I'm not sure which. And they have various geosites. I think there's 12 or 15 geosites here in Panama. And we've already sampled some last year. We will be sampling 3 different geosites this year using both malaise trap and UV bucket traps. We're going to be doing a project, which I'll show you on a later slide, on Rio Cherokee Viejo watershed. It's a major, one of the 52 major watersheds in Panama. It's the one that's immediately adjacent to Costa Rica. And last, we're going to be doing sampling of Coiba Island, which is a very interesting island off the in the Pacific, off of the Pacific coast of Panama. This is the Rio Cherokee Viejo watershed. Like I said, Costa Rica is immediately to the left or west of this. And we've set up 3 different routes at 3 different altitudes. Lowest altitude is probably 100 to 200 meters. The middle altitudes are probably 500 to 1,000 meters, and the upper altitudes are probably 1,800 up to 2,000 meters altitude.
Brian, just you have about 1 minute left.
That's fine. I'm about done. Thank you. So that's that we'll be sampling that monthly, 1 night per month, 1 route per night, and all the way through June, starting in February. Coiba Island is the last one that we'll be working on. This is an island in the Gulf of Cherokee off the coast of Panama. It's a former penal colony. In 2005, it became a World Heritage Site. It's been separated from the mainland for 12 to 18,000 years, ever since the beginning of the last warming period on Earth. And we expect to find some very interesting species. A lot of endemic mammals and birds and other insects are known from that island. So we expect to find the same sort of thing with the caddisflies. Finally, I want to reemphasize our purpose is not to count coup like the Plains Indians in the US. We're not into just describing species and finding new country records. We want to serve as an example for other people in other Latin American countries, particularly Central American countries, because a lot of our information is worthless without being able to compare with what people have in other countries. And right now we don't have that. So we'd rather other people get involved in other countries. And finally, if you're interested in any of our publications, having trouble sleeping at night, you can look on my ResearchGate site and download most of those publications. Thank you very much.
Thank you, Brian. Questions? Yeah, question from us? Yes, Brian, the email contact that is on the program is okay?
Yes.
Okay. Yes.
Oh, wait a minute, time out. You mean you're talking about the address?
Yes, your email address, I think they are asking if it is correct and if they probably can contact you on, right? Yes.
If you want to mail something or send something, send me an email, because unfortunately, Panama does not have mail service.
Okay.
That's our physical location, but there's no mail service. So if you want somewhere to send a letter or something, contact me by the email address that's on our program there.
Okay, perfect.
All right.
Thank you.
Brian, please stop sharing your screen, and you can mute yourself as well. I'll prepare the slides for our next speaker.
Okay, now our next speaker is Joe Martinez, redefining the notuides sensu stricto and reclassifying the notuides sensu lato. Your, where is the, oh, here. The stage.
Hello, everyone. This is Jose Martinez. For those that know me, actually, oh, maybe can you press?
Yeah. Yeah.
I'm the actual curatorial assistant here at the Aquarium Center. My focus is in Noctuidia, the entire superfamily of Noctuidia, working with evolution, systematic taxonomy, behavior, conservation. Again, everything just in case a position occurs in Europe. So yeah, so for those that know, Noctua is the largest superfamily of Lepidoptera. It has from 40 to 70,000 species worldwide. But the most important thing about Noctuidia is that there's only 2 families that are stable so far. The other 4 here they are mess, literally, and really huge. And everything started with this guy here, Noctuidia. That's why I started working with this group. So also I have a background in IPM, so that's why I've been involved with the mess in Noctuidia. Despite the diversity of Noctuidia, it's amazing that it's really monophyletic. So there's, you know, molecularly is proved that Noctuidia is monophyletic. But as a taxonomist, also I want to work with morphology, right? So there are 2 main morphological characters that we use to identify Noctuids. The first one is the tympanoid organs that are really easy to identify actually. They are in the metathorax and the first abdominal segment. And they can account this guy here, because this part here is the tympanoid sclerites. It's going to be useful for what I'm going to say later. Another important character is the Nigrobicetus that occurs in every single Noctuid except in the Acellular family. Well, after people have been working with Noctuids for a long time, they still have this problem here, and is that low support among 2 families. And this program has 10 years and it's still going on because this guy doesn't want to work on that. So obviously I have to do the skill for them. One of the things when I've been working in Lepidoptera in general, so we found for apomorphic characters, right? So this apomorphic character you can see in every single family, like we know that this is a species based on the caudal spine, you know, the huge bodies in Saturniids, the hairs in Lasicampis, the looping locomotion in some, well, mostly of the geometrids, huge heads in Hesperids, and the modified prolex in the family, right? But what about in Noctuids? It's impossible to find something that say, well, this guy belongs to this guy, or this guy, or this guy here belongs to Noctuidia, right? So just because we don't have a more apomorphism. So it's very difficult for a student to work in the entire family. So basically what I did was looking for something that has been classified as a family before, but they place it as a family just based on molecular data. This is a small group, Pentatians, a really amazing group, I have to say that. And there are a lot of morphological characters that separate Pentatians from the rest of the Noctuidae. For those who don't know, so it's a really small group with just 2 tribes, 4 genera and 150 species. 33 of the genera are census territory, right? So that's low. But it's still digging into these problems. I found that probably the monophylum of Noctuidia is not good enough. Why I say this? Because this guy here was separated from the actual Pentatians just because he's colorful and is diurnal. So, which is I don't consider this a good character to separate a type from another. The other thing that I was seeing is that they put the genus Thylacides, that's the only genus in Thylacides, as a synonym of Penthiaes. And the problem with this is that all these guys, all the Penthiaes are related with high elevations on very extreme areas. I have to say, deserts, xeropid areas, and these guys are totally tropical. But we can say that it's actually, part of the competition. So that's why they changed the habitat, right? But they asked for this specimen, this guy that was in names as a Thylacides sp. It was the genus Carmina. It's actually a Penthiae. So that's why I started making more conclusions about this stuff. So based on that and that information, I have to create my first phylogenetic analysis using 64 Penthiaes for a Noctuids belonging to other families not related with Noctuids, and 2 specimens just to corroborate that the actual placement of this, the new proposed clades are in the right place, right? So I did the maximum likelihood analysis using IQ32 and the species convergence analysis like we usually do in the lab. But again, as a taxonomist, I like to work with taxonomy. I used 209 Penthiae species, which, well, I consider species. Because I remember before it was 150, but after I was working on them, I found more species that were close related. So, and I could identify 86 morphological characters. The craziest part for me was that as much I was getting into the Penthiae, more morphological that I was getting. The most important part for me was that the tympano organs are unique for the clade. They're always the same thing, despite the they have different sizes. The shape is exactly the same thing. It looks like a small hair comes, all of them. And that was the first thing that I started using for this analysis. But also, people will say, why you don't synonymize all these groups in the same Penthiae, right? Because I got this guy into the play. Cuculiae is impossible to synonymize in Penthiae because multi-Penthiae, they have hairs and everything, and this guy doesn't have hairs. But I also found that the morphological characters in larvae are more important than the adult characters. So that's why I decided to say that probably it's not a Penthiae, but Penthiaes. So how we know that? Because after I finished my first approach, I got 13 subfamilies, 24 prides, 4 genera, about 400 species. My first classification. So then I start dividing the groups on these 2 terms that I create, Eviformia and Noctiformia. All those guys that looks like aerobites and have elongated body in larvae, obviously, and compressed body in Noctuids. Well, I showed this to Akido, so Akido was impressed with that, and he gave me the opportunity to get more data. So thanks to that, I was able to get more data from all the subfamilies involved in Noctuidae, including more data for other Noctuids, the 3 species of Macroteracera to create the actual trade proper way. And I had 2 butterflies just to see the placement of the new taxa I'm proposing, right? Obviously, I have more morphological data from other 309 species. What I found is that I could separate the 3 Noctuids from the rest of the Noctuids and Sulago. They end up being 5 new families. So the first one is Diphtheridae. Diphtheridae was placed in knowledge for a very long time, just based on the case that they are using a boat-shaped cocoon, which is not a good character because they've found this in Italy as well. Again, the all the characters of the temporal organs are unique for this group. The second one is Eustralidae, all this, the entire group is all these families were placed in the same subfamily before. So I don't know for what reason they decided to change this, just based on the data. But all of them have really significant important characteristics you can actually use. The only one that's creating a lot of noise is this guy here, Biodiversity, because they have fused bonds on the base. It's something that Jim Hayden was talking about. The morphology in this guy in genitalia is really crazy compared with the rest of the Entomology. So that's why and the support for this clade actually with the rest of the Stratigrams is really different and very low, I have to say. That's why probably they are different thing, but I don't have enough data. So another one is this Phthiraptera. So Phthiraptera, it was amazing way that I start thinking about this group, because I have this equation with one of my colleagues and Paul Gomes in the Smithsonian. For years, he was saying that this is a different family, and he actually published a small paper that saying that probably these guys are a different family. And here we can see that this is a different family. There's a lot of morphological characters that separate in this group from the rest of the groups. And that's why it's really easy to identify. Still, we have exactly the same problem with Cosmopolitan that we see in biodiversity, and it's because despite they have some of the morphological characters, they still are low supported, right? So this is the new one. So I love this one because 90% of the taxa here were pentatomoids. So all of them are tropical, all of them fits almost in the same theme, with the only exception of this group that fits on palms. Again, the morphological character for this one is the C shape in the tip of the stride. It's very easy to identify. There's no way that you can confuse these guys with other things. And finally, the true noctuids. So the true noctuids, the true noctuidae are these guys. Most of them you can actually separate them based on that. They don't have hairs or anything. They literally are naked. But for me, the most important part in the atlas is the claviformous pod is broken. So broken by the antemedial line, which doesn't happen in other clades, right? So, and the huge Phthiraptera. So it doesn't matter how big is your specimen, it can be super small, super big. You're gonna have this huge and massive Phthiraptera. So as a result of everything, I found that most of the people that be using the true-fine versus qualification in classifying noctuids, I don't like that character. So I found this because the true-fine donation, of course, also in the qualifying donation play, and something there's no good. Because it makes it very difficult to identify them at the end of the day. But in this case, it's really easy to identify that Eraceli and Notodon, they don't have tympanal spray, while the rest of the noctuids, they have tympanal spray. Here are two examples of what I'm saying, why this close relationship doesn't mean synonyms, right? So one of the things that we can see is that there's two true families here that they have 100% support, molecularly speaking. They are different families. Every single researcher, specialist in bombicoids, and they say that these are three different families. And the funniest part for this thing is that Arlen Gott was one of my friends that actually former student in our lab. And for years we were discussing about if seasonality is seasonality, and he say that it's not because they call this drag, the ground beetles. Forget we were discussing this. So at the end of the day, I said, man, they are different, and he never believed on that. So 3 years, 3 years ago, always this, and they changed against the actual classification. As a conclusion, I started scratching the surface of this problem. Honestly, I don't consider I fix everything. There's still some gaps there. So I could separate noctuid census data from noctuid census value easily. The close relationships with a synonym below. I propose 5 new families using molecular and morphological data. I'm reclassifying right now the pendians. Honestly, I advise the user of morphology to support phylogenes, molecular phylogenes. And obviously we need more work in that. Well, thanks. Questions? Well, I don't know. Yeah.
Yes, yeah. Any question? Do we have questions on Orzu?
So now Cicindelidae, it's a family.
The family.
Okay. Go back and forth.
Okay, thank you. Okay, our next presenter is Jose Marcelino. And his presentation is integration of omics approaches as molecular tools in apiculture research.
Thank you. Good afternoon, everybody. I'm Jose. I'm from Portugal, hence the accent. And I work at the Honeybee Research and Extension Laboratory, which is led by Dr. James Ellis. And we work with honeybees. So basically what I'm trying to present today is an overall approach as to how we integrate genomics approaches as molecular tools in apiculture research, but scaling up from genetics to genomics as technology evolved and as resources were available. We could scale up from doing analysis on fractions of DNA to whole genomics, whole DNA. So I'll start by giving a brief introduction as to the domestic honeybee, which is the central pollinator across the commercial pollinator across the globe. Just in the US, the almond industry is responsible for $9.2 billion with a B, $billion for the US GDP, and services that honeybees provide for the US are estimated between 12 to 50 billions a year. So there are many subspecies, and the migratory nature of beekeeping leads to a global dissemination of the species and all the subspecies. So there are 25 to 33 subspecies. There's not a consensus to some of them because some of them are reported from very isolated places like a mountain in Indonesia, and then we cannot have access to the samples. So we rely on most of the times very succinct morphological evaluations, which have proven not to be sufficient. So just to give you a background for the US, with the advent of steamship, there was a lot of unregulated movement of honeybees. And there were a lot of introductions. And again, they were not regulated. So there were a lot of honeybee subspecies from many parts of the globe, from Asia, from Africa, from Europe, from the Middle East. It was a free-for-all. As long as there was a market, there will be bees. This is from the early 18,000s, and there's things from Israel, from the one that they mostly, is this a pointer? Yeah. that still to this day is preferred is the Italian honeybees, Apis mellifera ligustica, because they have a high yield production and they are very quick to produce. However, the disadvantage is that they quickly go over what they generate. They eat a lot. But again, there was no regulation, so everything could be brought in. And then in 1922, the Honeybee Act was erected to legislate and regulate the movement of all of these things, because the different subspecies translate in different traits, and some of them are valuable, but some of them are not. Some of the subspecies might be defensive, they can outcompete the residents, all these problems that happen with invasives. But in this case, the movement was intentional and human mediated. So even if they are invasive, they were introduced intentionally. So again, this was until 1922, but it was already too late, because not all, but some of the species were already in the US, and then again, the migratory nature of beekeeping made the dissemination of these subspecies very quick. And this is from 2020. And this is not the total. I mean, it seems like there's where is the pointer? Oh, it doesn't show the pointer in the white. Well, it looks like there's like a temperature limit, but there's not. I mean, it is for some of the subspecies, but in the north, the Ligustica and the C lineage, Carnica and Ligustica, and the M lineage, which is European, are also present. It's just that this is not completely updated. But again, they are not pure. So because there's so much hybridization, people say, oh, I have a queen that this queen that I have is very good because it's resistant to varroa or something. And then I say, oh, but this one that I have is very good because it produces a lot of yield. So, they cross the drones from one with a queen from another one. So there's a lot of admixture, genetic admixture. So it's very difficult to untangle all this subspecies complex. And just to give you an idea, at the US ports, air, land, and water, the USDA has a program to intercept not only bees, but everything that arrives. And for Apoidea, 82.9% are all honeybees. And if you see here on top, they mostly come alive. And actually, I don't know if you know, but one of the most relevant subspecies is what they call African honeybee. I say this because for me it's African derived, it's not African. But there's a lot of interest on tracing this before they established. In fact, they say that this Africanized arrived to the US via Texas, but in fact it arrived via Miami, but they intercepted, and in Texas they didn't, so it established. But the first in 1989 was Florida, and then it was intercepted. It was a complete swarm in a boat. And they intercepted them, they euthanized them. But in Texas, when they discovered them, they were already established. So this is very important to trace, because not only the subspecies, but also there are some species of honeybees that are we know from other parts of the globe that they are extremely competitive and they outcompete residents. For instance, Apis cerana, and there are intercepts of Apis cerana. This is 20 years of data from the USDA, and they have intercepted Apis cerana. If that one arrives, it will be very, very, very bad, because in other parts of Asia, it completely eradicated locals. So all of this might have a lot of implications economically for the US, as it had in other parts of the globe. So how many subspecies do you think there are right now? I mean, that we have data. We have a number. This is 33. Like I said, this goes from 25 to 33. Some of them, there's no consensus because like I said, we don't have access to the specimens. And you know, the one in Indonesia, I've tried to get specimens, but it's almost impossible. It's in the middle of a forest and they don't want to sample, or there's permits issues and all that. So we don't know. But how many do you think there are in, do you have a number? That's exactly 10. Yeah, there's 10. I mean, there might be more. It's just that, you know, it really depends on the genetics and the morphologics that you do on the specimens that you can trace. So what we need to do, and when I say we, it's the USDA and us as geneticists or taxonomists, is to have expedient methods to, you know, trace them, monitor their spread, and early detection, so we can detect them prior to their establishment, because then it's very difficult to eradicate them. You know, a queen can mate with a lot of drones, and then she will use that semen for many years. So it's you know, the use of the semen will be cryptic for us. I mean, the seed for the subspecies dissemination is there. We just don't know. So we need to trace them. So we need to monitor them. The ones that are already here, detect the ones that might arrive, and certify queen origin, because it's a big problem. You know, people get queens from all over the place, even if it's illegal to get them from abroad. You know, sometimes they put them in a little box, and sometimes they arrive alive. And then we also want to detect organisms of concern, which is pests and pathogens, before the onset of signs and symptoms. And before an economical threshold is reached. So all of this is to get to what we do with genetics. So scaling up, what we have done in the lab is we have done restriction fragment length polymerase. It's like a little pipeline that is quick and accurate to have in a few hours, you can try to know at least geographically where they're coming from. So you can do RFLPs, which is you just profile them, you get a barcode basically with enzymes you can cut with specific enzymes that cut at specific points of the genome. And then you can have a barcode. This one is for the species, and you can see the bars are different. So this is the DNA cut in different positions of the DNA. And then with this, you can quickly visually have an idea of what it might be. And then you can also, for the subspecies, there are many markers, but this one is also one that we try to develop for a quickly assessment of the geography of the specimen, the unknown specimen you might have, meaning, you know, if it has a C allele or a T allele, you'll have one cut versus two cuts. And if it's let's say one cut, it means it's sub-Saharan African origin. And then if it's two cuts, it means it's all other mellifera, basically in Northern Europe or Central Europe or Middle East. So then you do this, which is a visual, and then you can so you can visualize, then you can quantify, and then you can profile. So you visualize with what I showed, and then you can quantify them with probes to have peaks and see, okay, this is this allele or it's another allele. And then you do amplicon sequences to have the actual sequence to determine if, okay, you got oh, I forgot to say. So we are targeting a SNP. A single SNP means a single nucleotide in the sequence in a particular spot. In this case, I was, you know, we try to do everything with mitochondrial because we can trace the mother more easily. So this is a mitochondrial gene, cytochrome B, I think. Okay, so this is with one SNP to discriminate the alleles. So this is the European honeybee, the African honeybee, and then hybrids. And as you can see, they differ in this single nucleotide. That's why it's called single nucleotide polymorphism. So you can start with this, and then as resources are available, because as you scale up, you also scale up in cost and technology and machines. This one is very quick and you don't need a lot of resources. But then from this, you can scale up to a set of SNPs. So you can screen the entire, oh, I think I forgot to show you something here. Yeah, so I put this the QR codes that you can do the forms. If you scan this, you can see the papers that what I'm describing relates to. So this one was for the history of the introductions and the movement of the honeybees. Sorry, I forgot that. So this one is from our group at the BLAC. And like I said, we can scale from one single nucleotide polymorphism in a particular spot of the DNA to screen the entire DNA for an array of informative SNPs that can discriminate the subspecies. So in this case, we are rich in genomics, so which is everything. So you do the entire genome. So in this case, this study was done in South Africa because in South Africa, we have two subspecies that are of greater interest for the Americas, which is the Africanized honeybee. So the accidental introduction and dissemination, well, it was not accidentally introduced. So it was the South African Apis mellifera scutellata was introduced in Brazil intentionally because they wanted to improve their stocks, but then it was released. They say accidentally is the formal explanation, but anyway, so this is the holy grail to discriminate the Apis mellifera scutellata from Apis mellifera capensis, which is the only parasitic species of honeybees that we know of. So as this is just capensis and this is scutellata, and then there's a hybrid zone between the two of them. Okay. And then we scale up for whole mitogenome sequencing to discriminate them as well. And I just wanted to show you that this requires a substantial amount of samples, and we have 85,000 from 47 countries in collection. And then lastly, sorry, I know I only have 1 minute. We wanted to do environmental DNA, which is metabarcoding of meta-transcriptomics, which we also do, to know what if we can find pathogens of these honeybees prior to the onset of disease, signs and symptoms. So this is varroa, small hive beetle, the wax moth, nosema, and foulbrood. And we scanned everything and we sampled all the surfaces and all the surroundings in the apiary to see if we could get this prior to the onset of the signs and diseases. And this is how we did it with the filters and swabs, forensic swabs. And we found everything except nosema. So this is valuable to find, to have a yes or a no for the presence of pests of pathogens without the visual observation. So before the onset of signs and symptoms. And that's it. So I wanted to thank everybody. This is a cooperation with many institutions, including FDAX and many formal researchers at the BLAB. So, and of course, funding agencies. So thank you, all of them, and all of you for listening.
Thank you, Lisa.
No questions.
Questions? One little one?
Yeah.
How is capensis parasitic? What's the behavior that makes it parasitic?
It's teletalking. It's so the workers can develop functional ovaries. So the queen can trigger with pheromones that they will develop the ovaries, and then they will go to colonies of the other subspecies, the lata, and they will lay eggs, and then the other ones will take care of those eggs instead of taking care of their own. Yeah, and it was a big problem in some parts of South Africa. Thank you.
Thank you. We have a last but not least talk. We have Bert Coleman, a Costa Rican biological puzzle. Is it over Zoom?
Yes, turning
Okay.
over Zoom. Bert, do you?
Can you hear me?
We can hear you. You should have permission to share your screen. Show us your slides.
Okay. Can you hear me and see me?
Yes, we can.
Okay. Yes, good afternoon. My colleagues, Angel Solis and Guillermo Alvarado, and myself would like to make this presentation, a Costa Rican biological puzzle. And what we try to do is to try to explain Costa Rica's incredibly high biodiversity using not so well-known information like geology, landscape, riparians, history, paleontology, and many other, let's say, not so very known pieces of information. Okay, let's see. I am presenting here the work team. This is Guillermo Alvarado, Angel Solis, and myself. And if you ever go to Costa Rica, any respectable restaurant will serve you your beer glass with ice. This is a reminder of the past where there were no refrigerators in the country. Now we have refrigerators, but the custom of still having ice in the glass is still done. So you should expect this whenever you come here for a visit. What we're going to present here is mostly going to appear as a chapter in the following book, Geomorphology of Costa Rica, during this year. It's being edited by Springer Verlag and edited by Adolfo Quesada and Piotr Migonia. Well, in order to try to explain the incredible biodiversity of Costa Rica, one of the main points we have to take into account is its orography. The country is incredibly mountainous, and the mountains have appeared at different times during the formation of the country, all this adding to the incredible biodiversity that this country shows. These mountains are incredibly important because they are cloud generators. You actually have the trade winds coming from the Caribbean and hitting the mountains. And whenever they hit the mountains, they generate all these masses of clouds, which of course make the country really humid, with the exception of this area where there is an orographic shadow. There are other areas that are very special because they generate around clouds. They have a system of rotor winds, very, very interesting. We can talk about that some other time. And we can see here cloud formation that represents the intertropical convergence zone, which is also a very important system for maintaining humidity in the country. Looking at climate, you can see here that the country is incredibly variable regarding all the climatic characters: temperature, rainfall, and precipitation seasonality. And the four main climatic factors that are controlling the climate in Costa Rica: orography, latitude, being a land bridge, and of course, intertropical convergence zone, better known as PH. Now, Costa Rica actually has lots of different types of vegetation, which you can see here. Costa Rica is actually the most biodiverse country at the square meter level. The country actually has more than 9,000 species of angiosperms, 1,640 orchids. And if we compare it with Colombia, which is the second country with the most amount of species per square unit, you can see here the enormous difference. This is important to stress because very many taxonomists think that Costa Rica is just one single unity, uniform unity, and this has made them commit lots of taxonomic errors, believing or thinking that everything is the same thing. And there are also problems with biogeography, where there are still many biogeographers that still insist at having the tropical dry forest stopping exactly at the Nicaraguan Costa Rican border. I won't say names, but this is not the case. The country is much more diverse and complicated than what many people think. It's also general wisdom or accepted wisdom that the rainforest is the most biodiverse ecosystem. this is not so. This is not the case. It's an analysis where you have here the rainforest and you have here the cloud forest. The cloud forest in Latin America will always have more species than the rainforest, not very much more, but still more than the rainforest. And the cloud forest will also have more endemic species than the rainforest. So this is something we have discovered in the last 10 years, and it contradicts basically all ecology books. Another thing that we also studied a lot was the conformation of the Costa Rican area during geological times. There are several maps showing how the country looked like during those ages, and some of them are really quite fantastic. So we actually studied all the information available up to last year, and we produced these two maps showing how Costa Rica more or less got conformed from the Miocene to the Pliocene. A very important point that has been debated all over is exactly the timing when the Panama portal got closed. There are many discussions, lots of information, but the majority of the people now agree that around 2.8 million years ago, the Panama portal closed. But as important as this, and not very considered, is the time when Costa Rica actually connected with Nicaragua, and the Nicaraguan portal got closed. And according to our studies, that happened sometimes between 5.8 and 5 million years ago when this portal was closed. And this date seems to be supported by the findings here at San Gerardo Limoncito, where the mammal fauna at this area is basically comprised of North American elements, not South American elements. So apparently, the northern elements got a little bit of advantage at invading insular Costa Rica a few million years before then the Central or the South American elements could actually do it. Important is this area here, which corresponds more or less to what is now called the Cordillera de Talamanca or the Talamanca Range. And you can see here that during the Pliocene, this area basically stopped having any volcanic activity. This is going to be very important, as we can see later on, or we shall see later on. Talamanca actually stopped being under volcanic influence because of the Cocos volcanic submarine range, which collided with the Costa Rica-Panama plate. And this submarine range actually subsided and started uplifting Talamanca at an incredible speed about 5 million years ago, separating this tropical area into two different areas, a Caribbean one and a Pacific one. And this fact actually helped create an enormous amount of biodiversity, as we shall see now. Since this area was basically an island, perhaps some traces of this past biology can be found. So we studied the genus Canthidium, which is a South American dung beetle group. And we invoked Foster's rule or the island rule, which says that organisms living on islands either tend to become very small or either tend to become very big, depending if the amount of food is limiting or if the predators have disappeared. And in this case, we found that the endemic species, which are these ones here, are actually statistically different from the non-endemic species coming from South America or from Central America. So Canthidium seems to be supporting the idea that there was an ancient island fauna that became adapted to this area just and simply by adapting their size to very limited amounts of food. We also studied Ontophagus, which is the ecological equivalent and which invaded from the north, actually from eastern China. And we can see that there is no such difference in size. Another thing that typically occurs on islands is wing reduction, like this case here, Ateuchus halutaceus. And if this area was really an island, as our model suggests, then we could probably find some traces of that nowadays. And if we check this map here, we can see this enormous amount of different species that have wing reductions and are concentrated on the mountains of Costa Rica. This is actually in the whole of American continent, the area with the highest amount of dung beetles with reduced wings. So this again supports the idea that actually this area was in the past an island arch. Continuing and going back to the Talamanca range, we can see here that when Talamanca got uplifted, it separated the country in two areas, a Caribbean one and a Pacific one. And we can see that this phenomenon has actually been producing species pairs. Some of them living on the Caribbean, the other ones living on the Pacific. We can see here, using dung beetles, the different Caribbean species. These are the Pacific species. And this again gets repeated with fishes, with amphibians, with reptiles, with birds, separating them into two different areas and forming sister species. Interesting is that on the Pacific side, most of the species tend to be very dark in color, whereas the Pacific ones, the Caribbean ones tend to be lighter in color. So this is apparently some type of logos rule. We can see here an analysis of the origin of the phaneus. And if you take the Costa Rican species, which are the red ones, we can see that this speciation more or less coincides with the formation and the elevation of the Talamanca range. So mitochondrial DNA analysis support this phenomenon as well. At the same time, a very nice and interesting barrier is formed here along this river, which is the Tarcoles River or the Tarcoles Line, whereas many species just and simply do not cross this river. In a previous analysis, we saw that there is a very great ecological coincidence in this area where different ecological variables change rapidly from north to south. But using our latest studies, we have found that these areas actually, or this river actually, helps the pyroclastic flow. So every time that the volcanoes have been producing lava, all this lava and the ash and the hot gas just and simply flows along the river exactly into the sea. And this has been happening at these times exactly. This pyroclastic flow can flow at more or less 500 kilometers per hour. These are the temperatures they generate, and this is the thickness they can actually produce. So you can see that every time that you had a pyroclastic flow, basically the whole area would be completely sterilized.
We have about 1 minute left.
And looking into the future, we can analyze the paramo vegetation, which is the alpine vegetation of Costa Rica. This is very important. It's the northernmost distribution of the Andean paramos. They are completely dominated by Andean elements. And we produced a model, which is the model that simulates a 4.5 degrees centigrade increase. It's called originally the doomsday model. Nowadays, it's called the business as usual model. And this model shows you how in 100 years, the greatest extent of paramo is actually going to disappear in Costa Rica and in Central America. So we have been trying to alert the population and educate the population about what is going on. So we have been publishing papers in the newspaper, trying to inform the population about this situation. And with this, I finish the presentation.
Thank you very much. Any question? Yes, Juliet.
Is there anything they can do to save the pheromone, or is it hidden?
Could you hear that?
No, I didn't. If you can repeat it louder.
There's a way to save the pheromone?
Right now, there is no way anymore.
No.
The only thing would be just to go there and try to collect species and try to keep them in greenhouse or something like that. That's a project we're trying to convince the government to do.
Thank you. Okay, so this was the last talk because we don't have enough time, unfortunately, to have the additional talks. We hope to have you on, Andrei, next year so you can give this talk. So I want to thank everyone for attending the conference, all the people in person and also over Zoom. We had a lot of topics, very interesting topics, so I hope you enjoyed it. I encourage you to attend next year. We'll probably have our conference in February and it will be hybrid. So you are free also to attend over Zoom. There are different ways to get involved with the organization. You can become a member, of course, and you can also publish on Insectamundi, and you can add with the conference if you want, and you can also add with editing for Insectamundi, if you prefer. For those that want the certificate of attendance, can send an email to Julieta, right? So they can receive the certificate. What I forgot. Okay, for those who want to stay for dinner, we are meeting at 6. And you don't have to leave because we have also the group photo before leaving. So, right, Julieta?
As best as we can.
Okay. So I want to thank Julieta because, yeah, she helped a lot in organizing this conference. I want to thank David and Oliver because they helped a lot over Zoom. Thank you. I want to thank Davide because he helped me as a moderator.
Thank you, moderators.
And yes, it was an amazing journey. So I hope you enjoyed it. And I hope to see you next year. And yes, that's it. Thank you, everyone.